Good morning, everybody, and thank you all people that are in this room for abandoning the coffee station and coming in so promptly. We appreciate that start to the morning. Welcome to the Polyphagous Shot-Hole Borer National Stakeholder Workshop. We have a number of you here in this room. For the people online, there's about 50 people in the room at the moment. And for those of you that's present here at the University of Western Australia, I'm trying to ignore the fact that we've got about 300 people joining us online today because that's quite a terrifying number. For everybody that is online and for those of you that are here as well, we do have technical support in the room, so nothing will go wrong with the online system, I am absolutely sure. In the case that it does, I have technical support. Caitlin and Josh at the back will make sure that we're back online straight away. So if there is any problems, stay online. We will be back with it as soon as we can. My name's Jen Garlinge. I'm with the Department of Primary Industries and Regional Development. I'm your facilitator for today. That does mean that I will be wrangling you on time. We've got a lot of speakers to get through today. We've got a lot of content to cover. And for the courtesy of those speakers, making sure that they get their allocated time and also making sure that the people online are able to join us promptly, I ask that you do observe those restart times for us. There'll be plenty of time to mix and mingle during the morning tea, afternoon tea and lunch. Take the opportunity for that, but please, when I come around, the restart times will be taken seriously. To begin with, I'd like to acknowledge the traditional custodians of the lands on which we meet today. For those of us here at the University of Western Australia, that is the Whadjuk Noongar country, on the shores of the Derval Yarragon, the beautiful Swan River that we've got just outside the door that we can see when we stop for our lunch break. I pay my respects to the elders past and present and recognise the enduring contact that those peoples have with this land, the waterways and the surrounding areas. I'd like to pay my respects to elders past and present and their continuing connection to that country. From the housekeeping perspective, if there is the need to leave this room, please evacuate through the emergency exits and follow anybody wearing a red warden cap. They will take us down to the Riley Oval, which is just north of this building. Please try to stay together and meet as one group down on that oval. And for courtesy again, although we will be asking you to use your phones to access Mentimeter for the questioning session, please put those on silent and try not to be overly distracted about them during the day. If there is anyone here that represents the media, we do have a media liaison officer with us today. So if there is somebody from media, Megan sitting down here will be able to help you with any other questions or any other information. Throughout these sessions, I'm sure you will have a lot of questions. There's a lot of information to be covered today, from information about the shot-hole borer itself through to international experience. We've got 2 international speakers present with us today, and someone will be joining us online later this afternoon. And we've also got information about the programs that are happening within the Transition to Management framework. During the day, you can ask questions using Mentimeter. For those of you in the room, you will have received this QR code in your folders. Scan that QR code and that will take you to Mentimeter, and you can add questions into that during each speaker's talk. And I'd recommend doing that shortly. There is a start-off question asking which area, which stakeholder group you belong to, and that gets us into the system. Those questions, if you're in the system, you'll see the questions that have come through from other people. You can like those if it's a question that you would like to support being asked. All those questions will be collated. Caitlin down there will make sure that those come to me for the Q&A session this afternoon. I think that is more than enough from me. So I'd like to get the formal sessions underway for today by introducing Dr. Vincent Lanoiselet. He will introduce the topics for the day, and then he will also follow up by giving you the story of PSHB in WA so far. Vincent.
All right. Good morning, everyone, and thank you for attending the workshop today. The workshop today is made of a lot of talks and discussion, and we've got great international speakers joining us today, and we are really grateful for them to be able to join us face-to-face and also online. Something that is also quite really interesting with this workshop is that we have a lot of stakeholders within WA and also across the country. That's why we have so many people online. So the challenge for organizing this workshop is obviously to make sure that we address the need to all those different stakeholders, both within Western Australia and also within the other jurisdiction across Australia. So a big welcome to everyone, and thank you very much for making the time to join us today. First, I would like to reflect on the response, the start of it, and we'll have a talk about it later on. But basically, since the first detection of the Polyphagous Shot-Hole Borer back in August 2021, it's been already 55 months, so close to 5 years. And by the end of the transition to management program, which is scheduled for December 2026, it will have been 5 years and 4 months. And that really highlights the fact that nationally, we have to, unfortunately, respond to incursion that are more and more expensive and that are not short-lived, but that become longer and longer to deal with. And that's something I really would like to highlight. It's been a lot of effort from obviously the DPIRD staff, but also from our colleagues from the other jurisdiction, from industry, especially the industry peak bodies, which have a say in the response, and obviously with all the stakeholders, such as the local government that have been impacted, the landowner within WA that have been impacted, and the local industry, which sit outside of the national response framework, and therefore that's been really challenging for us to be able to explain what's been happening and take everybody along our journey. Usually when we respond to incursion, it's incursion that affect industry only, whereas PSHB has been affected a huge range of stakeholders, and also we are working within an urban environment. So a couple of statistics that I would like to highlight to remind people about the sheer volume of work that's been happening since August 2021. More than 3 million trees within the Perth metro area have been inspected for the presence of the borer. More than 5,000 trees have been removed due to the fact that they were infested with the borer. Many more of those of trees were pruned because obviously if we didn't have to remove a tree completely, and we could get rid of the infestation by removing part of the tree that was infested, that was the preferred option. We had more than or up to 120 staff in the field every single day when at the peak of the response, and overall we had up to 187 staff involved in the PSHB response alone. To put things into context, that's probably about 15% of the size of the department. So we had many people such as planning, operations, surveillance staff, obviously laboratory staff, communication staff, media teams, legal officers, compliance officers, and many others contributing to the response. Overall, more than 1 million kilometers have been traveled, and I'm glad that this didn't happen during the fuel crisis that we are currently experiencing with high prices. Over 12,000 samples were collected and submitted to diagnostics, and more than 200,000 traps were deployed and retrieved and sent for analysis to our laboratories. We've learned a lot with this response, and over 130 host trees have been added to the global host list of PSHB. And that's a list that is being maintained on our DPIRD webpage, and that will keep on being updated as we gather more and more information about how the pest is behaving within an Australian environment. Unfortunately, several native Australian and also Western Australian species were found to be susceptible to PSHB. I also would like to reflect on the great support that we've received from the pest community, from the local government, and from all the other stakeholders involved as part of the response. So today will be the opportunity for us to hear from response staff and also from international researchers and also from the research that has been allocated as part of the $2.17 million that has been allocated by the Western Australian government on top of the cost sharing money allocated to response to this incursion. I also would like to sincerely thank our international speakers, Professor Shannon Lynch from the University of California, Professor Francois Roets from South Africa, and also they are both with us today, and that's great. And I encourage you to talk to them during our morning tea, afternoon tea, and lunch break. And we will have Professor Hurley online from South Africa. And it's really great, and I'm really thankful that for you to come, all the way to Western Australia, and also Brett, for you to start really early, noting that it's in the middle of the night in South Africa as we speak. Also, and that will be my final point, thank you very much to the WA Agricultural Research Collaboration research partners. So that's Professor Trina Burgess from Murdoch University, Professor Theo Evans from UWA, is based here, and Professor Ben Phillips from Curtin University, who are going to, or who have already started working on the PSHB research within a Western Australian context. So having said that, thank you very much, and we will go through the story of PSHB in Western Australia. A lot of you, especially the people present in the room, will probably know about the story of PSHB. Some people online might not be as familiar as to what's happened since August 2021. So I apologize if I'm going to, for some of you, present some of the slides you've already seen. But as I say, we have to cater for an audience of more than 300 different stakeholders across the country. So what is PSHB? So PSHB is a very small borer. To put things into context, it looks quite big on the photo, but it's basically the size of a sesame seed. So we're looking at a small insect that is about 2 millimeters long by 1 millimeter in diameter. So it's very small. What is this PSHB so much of a problem? So it's basically a borer that is going to bore through a lot of wooden species. The very particularity of this borer is that it is not a termite. It's not going to eat the wood, but what's going to happen, it's going to farm a fungus. And so basically the borer's got very strong mass part that will be able for it to bore tunnels through the wood. And you can see on the photo the kind of tunnel I'm talking about. So it's creating those tunnels, and then it will farm a fungus, which basically it carries the spores of the fungus in its mouth. And once it infests a new tree, it will basically start tunneling and then farming the fungus. A little bit like a lot of ant species that are basically cultivating fungus to basically feed on. So the activity of both the fungus, which is a fusarium, so the activity of the tunneling and the fungus growing inside the vascular system is going to cause the problem. And it basically starts with once you've got this kind of damage within a branch, as you can see, there's a lot of tunneling, and remembering that the fungus is also growing through the vascular system. So at some stage, the vascular system is so compromised that the branch will start to die and fall. And eventually, when the borer compromises the whole trunk, then the tree can eventually die. So for people that may not be familiar as to what a vascular system is, it's basically the equivalent of a blood vessel within our body. So basically, if you cut our blood vessel supply or our artery, you can imagine what's going to happen. So that's a very similar situation within a tree. The vascular system is the vessels that basically will enable the movement of water and nutrient to go within the tree. So the first symptoms that people experience is usually a dieback within the tree with branches starting to die, eventually falling off. And that's exactly what happened in 2021, in August 21. For the context too, I would like to remind everyone that back in 2021, that was not quite the peak of the COVID pandemic, but that was the tail end of the pandemic. So basically, the beetle is also native to Southeast Asia, and it's been detected in Israel in 2009, Southern California 2023. So the Californians have the most knowledge about this beetle, especially in terms of management and research. Hawaii in 2007, South Africa in 2017, and Argentina in 2023. And it's been detected, obviously, in Perth in August 2021, unfortunately. So that shows you a little bit of a picture of where PSHB is known to be present in the world, and the fact that it's a beast that's moving across the country, the world, unfortunately. So the first report was provided by a member of the public. So someone in East Fremantle at the end of the COVID pandemic started to realize that two of box elder maple tree were starting to look sick and showing signs of dieback, and the branches started to fall. So that person knew about the My Pest Guide Reporter app, which is an app that was developed by the department quite a few years ago, and took some photos through the My Pest Guide Reporter app. Those photos automatically goes to the Department of Agriculture here in Perth, and the photos were looked by a diagnostician, and the diagnostician decided to send two biosecurity officers on site to investigate further just based on the symptoms. So as you can see, this is the photo of what the symptom looked like. Sample was collected and brought back to the DPIRD laboratories, and the result came back as Polyphagous Shot-Hole Borer, and that was the start of the response. On the photo on this side of the slide, this is basically the first quarantine area that was proclaimed. At the time, basically, we had only one detection of PSHB, and therefore we focused on a quarantine area that was encompassing the local suburbs, and we started doing the limiting surveillance across the Perth metro area to find out how widespread the pest was. So this is a slide to explain a little bit about the national arrangement as to when an exotic pest or disease is detected in Australia. So there is a whole framework, which is the Emergency Plant Pest Response Deed, with cost-sharing arrangement and government arrangement in place. So when we have a pest that is exotic to Australia or a disease that is exotic to Australia, we've got a whole system in place that takes care of the investigation. So the first step is basically that the combat jurisdiction has 24 hours to report a suspect exotic or a confirmed exotic to the Commonwealth. So that's the DAF Commonwealth based in Canberra. So the Chief Plant Protection Officer, is Gabrielle Vivian Smith. So that was done. And then the whole situation. So we provide a situation report so that the Consultative Committee on Emergency Plant Pests can consider the implication of the detection. So the first thing that this committee is going to be looking at, and basically that committee is made of all the chief plant health manager across the country. So that includes all the jurisdiction, all the state and territories, the Commonwealth, and also the industry that are potentially impacted by the pest. So here we mean the green life industry, the palm industry, the avocado industry, all those industries were part of the consultation process and the decision-making process as well. So there's three questions that committee is going to look at. The first one is that, is the pest really exotic to Australia, and is it going to cause any economic impact or any major problem with the environment and the like? Then, so if the answer is yes, the next question is going to be, well, is it technically feasible to eradicate the pest? And then again, if it's yes, is it cost beneficial or not to eradicate the pest? When all those questions are answered by a yes, and that's decided nationally, then the combat jurisdiction is invited to submit a response plan to eradicate the pest. Once that response plan is submitted and agreed upon by the Consultative Committee on Emergency Plant Pest, it goes to another national committee, which is the National Management Group, which is basically all the Director General across Australia, to make it simple, and also the CEOs of the peak industry body that are potentially impacted by the pest. And that group decide as to, yes, we endorse the response plan or not. And once the plan is endorsed, then the funding mechanism is agreed and the money is available for the response. In the meantime, it doesn't mean that the department do nothing and wait until that decision is made, right? It just means that we've got the mechanism down the track to recoup the money that we've already spent as part of responding to the detection. So this is basically just to highlight that the national decision and the cost-sharing agreement are nationally agreed within all Australian government and the peak industry bodies. It's not just a WA decision, it needs to be nationally coordinated and nationally agreed. So once we did the delimiting surveillance, we sort of detected the pest in several suburbs around the Swan River, and therefore the size of the quarantine area increased. And it went, as you can see on the slide, it covered 25 LGAs, so that quickly started to be quite big, 871 square kilometer, and that included 192 suburbs. For the people that may not be familiar with the population of Perth, we've got about 2.4 million people in Perth as present. The quarantine area at the time didn't encompass the whole of the Perth metro area, but a very large section of it. We're probably looking at somewhere 1.8 million people living within the quarantine area back then in October 2022. Response plan, $41.3 million, again, cost shared across all the state and territory and the Commonwealth. And therefore, a lot of activities happen in terms of the surveillance to find out the delimiting spread of the pest. So that meant inspecting trees visually by field officers and also trapping. So we had more than 3,000 traps deployed across the metro area and also outside of the metro area, obviously, to make sure that the pests were not present in the region or anywhere else in the rest of Western Australia. The response plan also catered for communication, containment activity, eradication, which is the treatment of the infested trees. So that phase went from August 2021 to June 2025. Because basically, as I mentioned, the response plan and all the information is regularly reviewed by the national committees. And a lot of work was done in terms of gathering all the information, providing intelligence to all the other jurisdiction and industry. And as part of all that work, there was a continuation of looking at what do we know now as the time being. Is it technically feasible to continue eradicating the pest or not? And so a lot of work has been done across the country, not just within WA, but for example, there was a lot of epidemiology work that were undertaken, fed by all the surveillance activities and all the information that we had gathered. And on the 18th of June 2025, it was nationally decided that despite all the work that had been undertaken in WA, it was no longer technically feasible to eradicate the pest in Australia. A national decision was therefore made as to what's happened now that the decision has been made. We basically can't eradicate the pest. And a national decision was made to transition to management. A decision could have been made as to, that's it, the response stops, and it's WA on its own now. There's no more national funding. Obviously, we had to strongly advocate, and a national decision was made that we would have 18 months to develop a transition to management plan that would remain to be co-shared to help us transition. And so that the response didn't stop abruptly. It just means that we switch from an eradication phase to a transition to management phase. And that happened in July 2025. And we were given 18 months of national funding to do that transition to management phase. And we will explain what we meant by that. So here I've got, again, a couple of stats available, which are already highlighted. But basically, when the decision was made, the pest has been detected on 169 properties across the metro area. So that's what I just talked about. On the 18th of June 2025, the national group determined that eradication was no longer technically feasible to eradicate. The decision considered the latest scientific data and advice from plant biosecurity experts. So we are 8 months within that transition to management phase, and there's 4 key programs or 4 objectives of the transition to management phase. The first one was to orderly stand down the eradication activities. We basically did that over 2 months because obviously you can imagine as of 1st of July, we still had some commitment. We still had made commitment to Stakeholder that we would be coming and pruning the infested tree or removing the tree. So when the decision was made, it didn't mean that, well, everything stops in terms of removing infested tree. So that's what we mean by orderly stand down of the eradication activity. We finished the work that we had told Stakeholder we would be doing. So that was the first objective of the response plan. The second one was to slow the spread of the pest, because obviously it's still fairly contained within the metro area, and the longer we can keep it where it is, the better off we are within Western Australia and for the rest of the country. The third component is the capacity building and training activity, which is going to be covered in the next talk. And the next one is the future proofing. So by future proofing, I mean what's going to happen at the end of the transition phase, and making sure that all stakeholders within Western Australia and also for the rest of the country are prepared as to the long-term management of PSHB. should it arrive in different parts of the country. So this is just a slide to show you the size of the current quarantine area. It covers pretty much the whole of the metro area. We've decided to create two different zones. We've got a management zone, which is in red on the map, which is basically the area where we know PSHB is prevalent and established now. And we've got the blue zone, which is a bit of a buffer zone where at the time there was no detection of PSHB or very limited detection of PSHB. And then we've got the rest of the state where PSHB is not known to be present. On the slide, something really important is that as of today, there's been no detection of PSHB outside of the quarantine area. It's still fairly contained within the metro area and still very much present in the management zone. That's my last slide. In addition to all the nationally cost-shared activities, the Western Australian government decided to allocate some extra money. The first program was a $7.2 million for the tree recovery program, which means that stakeholders that had the tree removed as part of the response for one tree, they can claim 2 new trees. And that's what that 7.2 million program is in place. And it's been run by the Department of Environmental and Water Resources, or DWAARC. Another pot of money was allocated for research, so $2.17 million was allocated through the WA Agricultural Research Collaboration, which we will hear about this afternoon. So that's research being undertaken under Western Australian condition. And finally, $2.5 million was allocated to support the local government that have been the mostly impacted by PSHB. So the government within the management zone or within the red zone on the map. Thank you very much. And we will go to the next speaker, who's Tom, I think.
I would like to start by introducing Robyn Boulet, who is one of the PSHB incident team leads and is managing the Slow the Spread program within the transitions management. Following Robyn, we'll have Tom Paul talking about the capacity building and training program. Robyn.
Good morning, everyone. Thank you very much for coming to today's presentation, and I hope that you get what you want to get out of it. So I am Slow the Spread Manager, or otherwise known as the Operations Manager, and today I'm going to be sharing some insights on the operational activities that are supporting slowing the spread of PSHB outside the management zone. So to give you some perspective, there's 3 objectives and 4 key activities that Slow the Spread I suppose, adhering to. So the first objective is looking at preventing long-distance spread of the beetle. So we know that the beetle is going to be spread through human-assisted movements of wood. The second objective is we need to reduce the beetle population pressures. So that is looking at establishing sentinel tree networks and how we can maintain oversight on those. And the third objective is regulatory activities that's aligned to the Biosecurity and Agricultural Management Act. So we're addressing these objectives by carrying out tasks or key activities. So that's through identifying high-risk sites and trees through the Sentinel Tree Network, performing surveillance in priority areas. So my team are out there undertaking a lot of surveying of identified trees. If we do find a tree, applying targeted treatments, and, we would consider things like the site, the tree species, and the infestation levels there. And the last activity is strengthening our compliance obligations and increasing awareness throughout the communities. Vincent has just touched on the quarantine area, but we are technically working in the containment zone, which is the blue area, and that is where little or no known infestation of PSHB exists. Of course, we've got the white area or grey area, which is the outside quarantine area, and that is where we know there is no, we're not aware of any known infestations of PSHB. So throughout the quarantine area, movement restrictions do apply. Green waste and untreated wood and living plants cannot be moved outside of the management zone, which is the pink area, unless they are covered and being taken to a green waste site facility. So that's a very key metric in managing the beetle. However, the materials can be moved from, or green waste materials can move inwards. So they can move from the outside quarantine area or the containment zone, which is the blue area, into the management zone. And of course, materials can be moved within each area. Slow the spread operations is currently focusing our efforts in the containment zone, and now we have just recently commenced doing some surveillance in the outer quarantine area. It's worth noting as well, the outskirts of the management zone, so the pink area, just within that boundary, there is, we are conducting some surveillance in the area. So that is sort of just on the inside of the border there. So and that is our way of ensuring that the Sentinel Tree Network program covers, you know, the very edges of both borders. Slow the spread structure really has three key areas. We have a total team of 40 staff, with the field surveillance being the largest group. So our schedulers work very closely with planning, who take instructions on areas of where we need to adopt mapping and reviewing of the Sentinel networks. Our field surveillance group, they are boots on the ground, so they are doing a lot of driving. It's a very big area that we're currently managing, and they're doing the surveying and mapping activities of the Sentinel Tree Network. And then we have the compliance team, who are raising awareness and providing guidance and issuing control notices under the BAM Act. So the Sentinel Tree Network is now inspected on a quarterly basis, and the Sentinel Tree Network actually acts as an early detection of possible PSHB spread. My team, or the Slow the Spread team, have mapped the most susceptible host species, including the top 3 hosts, and that being the box elder maple, the robinias and coral trees. These trees were selected on their susceptibility and accessibility for inspection, and were found in targeted locations of environmental and horticultural significance. So far, the Sentinel tree network, we have mapped over 2,356 sentinel trees, and with which is within the surveillance zone, which was the blue area, and 108 trees in the outside area, which is just on the other side of this red line here. The sentinel tree network spans more than 3,000 square kilometres. And in addition to the sentinel tree network, we have over 100 traps located in regional locations. To date, we have had one confirmed detection in the containment zone, which is in the blue zone, which is the area of little or no known infestation. When that tree was identified, it was identified through one of our sentinel trees, and a risk assessment was prepared, and that particular tree was removed. So the response was really agile and responded urgently, and reprioritised our focus to the affected area, where there was significant collaboration with stakeholders, local stakeholders and the wider community to increase awareness that PSHB was actually present in that local area. Not only, so I'm moving on now to compliance. So our compliance team do a lot of audits with green waste facilities, and those audits. So the couple of things that we've done recently is we've changed the audit criteria, and we have increased the duration, or the rotation, or the rhythm, I should say, of the audits to 3 monthly or quarterly. The audit, so that just commenced in January. We've got an audit starting next month, so I can't believe it's just come around so quickly. However, you know, the audit results that we do get, we assess those and we start working with those green waste providers on any non-compliant measures that were identified. Also within the green wastes, we've got 65 traps and at green waste sites and arborists. And I didn't mention the green waste sites that we are working with are both local government and privately managed. So we've got two quite different drivers, I suppose, for each of those areas. And lastly, where we're looking at identifying high-risk pathways for green waste to continue slowing the spread. So that is slow the spread in a snapshot. So thank you for your time.
And now I'd like to introduce one of the other PSHB incident team leaders, who is Thomas Paul, and he'll be running you through the capacity building and training program that is in operation at the moment as well.
Hi everyone, thanks for that. Yes, so my name is Tom. I'm the manager of the capacity building and training team. I came to the PSHB response after learning many important lessons about emergency response through previous roles, such as a pathology collector or as an administrator, especially during COVID. This included working at the border, the WA border, testing every entrant of the state. So yes, I was there when there was a truck explosion, a Telstra outage, and a rail outage all at the same time. I have also, obviously, I've got a degree in ecology and conservation biology and interested in plant sciences. So emerging pest management felt like the perfect fit. I'm incredibly lucky to have managed the team that I currently have, who bring a wealth of diverse experience, including specialized knowledge that they built working within the response. Many of my team have worked their way from entry-level field surveillance officers in 2021 to case managers, logistics, communications and planning officers, and they're now applying all their PSHB experience to capacity building and training. I just wanted to ask, does anyone know where the term capacity building comes from? Interesting enough, it first emerged in the movements for social change in the 70s. And it basically is about giving people the tools for self-determination. It's basically teach a man to fish rather than give a man a fish. So we're here to teach stakeholders how to fish in some sense. In this case, manage PSHB, acting as guides towards self-determined effective management. So why does CB&T exist? So in the response's transition to management in July last year, it created a need for consistent, practical and scalable training. Through collaboration with the CCEP and other government, nationwide government bodies like the NMG, we learned that stakeholders needed accessible and practical training to support long-term PSHB management. So long-term containment requires capability embedded in local organizations, industry, frontline teams and communities with a consistent and alignment across states. Our aim and purpose, well, it's an integral part of our nationally funded transition to management and aims to build capacity across Australia and support all Australians to prepare and to treat PSHB with consistent understanding and practices. So as I was saying, its purpose is to deliver targeted practical training to these industry partners, to local governments and community groups. And this is obviously across state borders. And so once we have the capability across these three sectors, we just strengthen our collective capacity for early detection, effective management and compliance. So who do we aim to support? As I said, local government, we've got a team that we've put together that that focus on these three broad stakeholder groups, such as industries, communities and local government. So just as like an example, this is obviously just a small list of the relevant stakeholders. This is specifically to industry. This is obviously where we've been in communication and engagement with agriculture, horticulture and landscape industries, as well as arborists and nurseries, and of course other state departments across Commonwealth states and territories. And this is the community group. Like I said, just a small number that we got started with. That we're obviously expanding on. And obviously local government, we have already managed to talk to, and provide training with our local governments within WAs. This includes the Perth metro area and regional towns. And as of late 2025, we've begun engaging with local organisations nationwide. Another unit within CB&T is our general information line. So all our questions about PSHB can be obtained through this general information line. This is one way to contact us directly, but apart from general advice, it can be for setting up training events, presentations, as well as assistance in using the My Pest Guide Reporter app for suspected sightings. And this is just to show that public information does still exist within our transition to management, and we work alongside them. So everything that we provide, PI also provide in their own way, including media releases, internal and external newsletters, and milestone announcements. This is just a rundown of what's happened so far with our team. So currently we've had 146 stakeholder groups engaged and over 1,400 activity attendants. This is just some small examples, but within them, so you have 40 activities within local government. That includes the workshops in the metropolitan and southwest areas, field-based training for PSHB detection as well. Industries, obviously, we've had their own versions of workshops with nurseries, arborists, and regional growers, as well as briefings. And with communities, this is just a small number of them. We've done many general information and Q&As for all the way down to presentations at and information sessions within universities. In the general information line, this is from last week, so I'm assuming we've probably hit about 100 1,000 phone contacts, and over 100 My Pest Guide reports have come through to us. This is just a dashboard of something that was created within our team. It's all captured. All our stakeholder engagement is captured in a customer relationship management system that we put together using the app Max that we call the Max engagement form. And this just tracks the interest, influence, and priority of each stakeholder. So it's just a bit of a dashboard of who we're engaged with at this point. So how do we build the capability that outlives the response and transition to management? An integrated pest management program has been developed and evaluated based on international and Australian experience and research. So this outlines available preventative and management measures across urban, natural, and agricultural environments, up-to-date information on pest distribution, and operational, strategic, and tactical information from DPIRD's Slow the Spread team. Within WA continues to provide valuable intelligence for the development of this. So the capacity building and training programs has developed and is continuing to fine-tune and develop a suite of training materials to support stakeholder adoption. In-depth consultation with stakeholder groups has assessed the most appropriate methods, resources, and delivery mechanisms for the transfer of knowledge and capacity building. This consultation has also assisted in identifying current knowledge gaps to ensure that the materials and methods produced are at an appropriate level for the stakeholder starting capacity. So the result of this was the creation of a national integrated pest management plan for PSHB. This has been made into a set of guidelines, and so all materials that's been developed from our team are drawn from these. These tailored materials will, by 2027 or by the end of the year, be distributed in packages containing whatever material is relevant to the stakeholder we are engaging with, and all approved CB&T materials will be added and updated on a dedicated PSHB webpage. So I'll just quickly run some through this. We don't have to read all of it, but essentially, obviously, we have a lot of in-person engagement. We have the trainer-trainer workshops for local governments at an operational level. We've also tailored to industries, so they obviously have a little bit of different information. Obviously, that's only relevant to them, and we have begun creating webinars of these. The workshops, just to quickly run in through what they are kind of at their bare bones, they're 5 core modules. These are very much based on the pest management plan and those guidelines. So we've got management planning, prevention and risk and reduction, surveillance and data, native and exotics hosts and sampling, and legislation, treatment and disposal. The native and exotics hosts, as an example, is based on the unique experience of the WA response. So notably the way that PSHB has interacted with our endemic host species and ecosystems in contrast to the international experience. And so these are from these core, we've created many different types of workshops depending on the stakeholder involved. And this will also include community groups. So yeah, more in person, I'll just run through that. So as I was saying, it's not just invitations to talk to us. We also go to annual general meetings and have been a part of university lectures, and obviously there'll be tailored presentations for all these. And the physical, so obviously we still work with public information. They have a lot of resources that they created. We obviously use those as well. And so we provide a suite of physical resources designed for real world field use. So using both the stuff that we've created ourselves, but also PI, we obviously have things like laminated ID sheets, checklists, treatment and disposal guides, and operational tools that crews can use on site. Our resource packs support frontline teams, such as parks, natural areas, nurseries, arborist inspectors, community groups, and contractors. And these materials are also integrated into our workshops, so we ensure that participants leave with practical tools that they can apply immediately. This is a new area within our within the entire program. This is the Integrated Pest Resource Suite. What this is based on the National Integrated Pest Management Plan, so those guidelines. This resource suite is being developed to provide a nationally consistent best practice set of guideline materials to support awareness, surveillance, risk reduction, and management of Polyphagous Shot-Hole Borer. So the best practice is based on research and observations and operations in the PSHB response. So they will be evaluated by different stakeholder types, and so this feedback will inform the final versions, making sure that we provide different types of stakeholders what they need. This is just a quick sort of mock-up of what it will look like. This one's very much based on for local governments, but they will be tailored to the needs of the stakeholders. And just so digitally, obviously everything I just said about the resource suite and as well as well the webinars as well. We have everything will be eventually digital. So we have e-learning modules that we'll begin working on. This will we will also have short video series. The webinars will be recorded and hopefully uploaded as well as the materials of the resource suite, all onto the DPIRD PSHB website that a portal can take you to the dedicated web page. And this is just a quick mock-up as well of what the web page will look like. It's very much like the Varroa Mite website. So it'll be an easy to use web page where you can go to monitoring, reporting, how to manage, training resources, and everything that we've created with our team and news, etc. And lastly, I just wanted to point out that, yes, I don't mean to be a broken record, but evaluation and consultation is how we are creating these materials, and that feedback is incredibly important to us. So it's not just within the when we do, say, presentations, Q&As and the workshops with our Mentimeter, we also do surveys to make sure that we are actually continuing to have to provide best practice up to the end of transition to management. And lastly, how you can help. You can by using our CB&T materials that we can support consistent PSHB management across your organisation, promote consistent messaging, and so contact at any time if you need clarification, share feedback as always, and support best practice guidance by integrating our tools, and get in touch with us because we welcome training requests, questions, and collaboration opportunities. And thank you very much for your time.
I'd like to present Dr. Kristy Moynihan, who is the program lead for the WA Ag Research Collaboration's Resilient Agricultural Futures program, and she's been integral in driving the development of the collaboration's PSHB FD research program. Kirsty's more than 20 years experience in research, strategy, governance, and evidence-based policy setting in various leadership roles within DPIRD and other WA governments. Kristy is passionate about achieving collaborative and innovative solutions to agricultural challenges, and she's here today to talk about the 3 research projects that have been stood up under WAARC's auspices. Kristy.
Thanks, Jenny. And yeah, good morning, everyone. And I'm very glad that I'm coming just after a break rather than just before it. So I think that's worked out perfectly. So first of all, just to introduce who the WAARC is, who we are and what we do. We are, as the name suggests, the collaboration of 6 organisations. So 3 WA universities, Curtin, Murdoch and UWA, DPIRD, CSIRO and the Grower Group Alliance, which is a network of grower groups throughout Western Australia. So as people have mentioned, I think Vincent mentioned before, we are a WA state government funded organisation, and we were invited by the Minister for Agriculture to contribute to developing up a series of research programs and to fund them. So of course, being invited to do so, that's what we duly did. So I'll give you a bit of an outline of what we did, to sort of get that research program organised, and then I'll go through and briefly give you an overview of each of those projects as well. So first of all, we developed some research themes. So this was in collaboration with a whole bunch of different stakeholders. So it's probably people in the room who contributed to getting that research framework set up. State and local government, obviously, also some academics and some industry folk as well. So we're very lucky to kind of get all that buy-in right at the start. So this was back in September 2024. We got the framework together, and basically there were 3 priority themes that we agreed on. So firstly, the host pathogen and environment interaction. So understanding the foundational biology was really important. Obviously got a beetle and a fungus and a host and an environment that is slightly different from where they occur elsewhere in the world. So understanding how those things interact in our environment and with our hosts in Australia was really important. Secondly, looking at innovative control strategies, so looking at chemical and biological controls to deliver scalable tools within the WA environment as well, or the Australian environment. And then thirdly, looking at detection and surveillance, so looking at new approaches to improve detection efficiency and decision support. So this is just, an outline of those, summary of the 3 projects that we did end up funding. Obviously went through, a process of receiving proposals, and then, sort of each of the 3 proponents that leads each of these came from the universities. So Murdoch University, University of WA and Curtin University. And I will talk a little bit more about those projects, obviously hoping I do justice to the research projects. So each of those are around similar sort of funding amount, around the 700,000, with a total value of just under 2.2 million. So in terms of, the industries and sectors that will, hopefully benefit from this research, we've got sort of a pest that is indiscriminate in its approach, obviously doesn't care who owns the tree, but, operates both in an agricultural environment as well as within sort of local government authority areas. Forestry is obviously also, there's concern amongst there because it could certainly affect forestry trees, the natural environment as well. We've got some very fragile and very precious wetlands that we'd like to protect. And then, of course, horticulture, so avocado, palm fruit, mango, macadamia, etc. So all of this information, while we are obviously a agricultural research collaboration, it will certainly help, in terms of control in these other environments as well. So outcomes anticipated in sort of a very broad sense, we're looking to improve management options and treatments for WA. And, as was said earlier, looking to slow the spread and reduce the impacts. We want to enhance early detection. So when and where this occurs, again, in different areas, we want to be able to get on top of it quickly. And, that helps because you can prioritise resources, knowing when and where to look. The other part of this, of course, is to increase the biosecurity research capacity and capability in WA. So each of these research projects includes PhDs and postdocs. So it's really, helping to build that next generation of researchers as well. So the first of the 3 projects is led by Professor Ben Phillips from Curtin University. So decision supports tools for managing PSHB is what Ben's looking at. So he's really, he's a modeller. He looks at how to kind of combine known information with, or some of that published information with the type of information and the data that we're collecting from the response in the field, and looking to, optimise surveillance through that. So looking at the predicted impacts and, obviously when and where to look for it as well, and in what species it occurs. And then he will be able to use these techniques in scenario testing for detection and different control methods as well. So integrating this existing knowledge, as I said, there's some published information about the borer that can be applied to models. So it's often temperature dependent. These sort of transitions between the life stages of the beetle. So at higher temperatures, they transition through those life stages faster. So what Ben's team is doing is applying that. So, sort of looking at a temperature map of the country, if you like, different time stages. And then from that, then developing, when and where it's likely to occur in higher numbers, and also, become more of an issue because that population's increasing. So just on the far side there, you can kind of see that resulting map that does very much look like the eastern states of Australia are quite exciting in that, probably that's going to be a place that shot-hole borer will really enjoy living if it ever gets there. So that yellow, kind of being that high-risk area. This hasn't had the host information applied to that map yet, so just keep that in mind as well. Obviously, the centre of Australia, where there are very few trees, if any, is not going to be a very conducive environment for those borers. And then just, Ben's team there, he's got some wonderful people working with him, and an as yet unknown postdoctoral fellow. Second project is led by Professor Trina Burgess from the Harry Butler Institute at Murdoch University. So Trina's team is looking at an integrated research program to support long-term management of shot-hole borer fusarium dieback in WA. As we said before, WA has got a really, quite a unique dynamic with that host pathogen and environment interaction, and it needs tailored solutions. So the project spans prevention, rapid response and long-term management, and she's looking to deliver science-based tools for this PSHB fusarium dieback in WA. She's got a team of forest pathologists, ecologists, obviously people with, sort of a real bent towards that natural environment. But as we said before, this is the information that we need in order to manage the pest at a landscape level. So, a couple of what I think are fascinating photos showing what actually happens in the lab. So, the first part of this process is to kind of look at the fungal biology and host susceptibility, and they do that by soaking some toothpicks in, a nice concoction of fusarium, and then inserting those toothpicks into some excised stems. So you can see in the lower photo there what that looks like when they're actually, they've been allowed to infect the branch, and you kind of get that brown staining around them. So this is what people do when they're kind of looking at them in a lab, and have had some kind of interesting early results from that. So it'll support management by informing that host-based surveillance and modelling portion. Another aspect of the Murdoch project, in collaboration with WA, they're looking at lures and repellents. So the first phase of this is developing some colonies of shot-hole borer using an artificial diet. So, sort of a series of photos there that show, that they've actually been able to successfully develop a colony of shot-hole borer, and that by feeding it a lovely sawdust smoothie type concoction there, that they're able to successfully establish in the lab environment. And then, sort of you can see that kind of fusarium development as well down the end there. So another part of the project that Trina's leading is in combination with a private company known as Arboriculture. They do some wonderful multispectral imaging, and you can see from the image there, to that degree of resolution that they're able to get. So a real canopy level resolution that can monitor tree health over a period of time. So that can, sort of give a bit of an indication as to which trees are showing signs of stress, and therefore you can kind of go and look more closely at diagnosing what's causing that stress, or parts of the canopy dying. So they do that. They pick up those images using fixed wing aircraft. It's obviously a lot higher resolution than a lot of the satellite imagery that's available. And the outcomes that they're looking for is obviously detecting symptomatic and asymptomatic trees, and to develop some dynamic risk maps and look at treatment impact as well and the monitoring of that. So doing it in conjunction with a lot of stakeholders, and there's probably representatives from some of those local governments in here today, DBCA, Kings Park, etc. So yeah, a lot of people involved in that, and I'm sure it will yield some really interesting work. I should mention also that while that's kind of, looks at tree canopy in the urban environment, of course, it can also be done at that sort of orchard scale, if you like. So these are some images that demonstrate what that looks like as well from that orchard scale. And Trina's team, this is only part of her team. She's got a number of other people that are involved as well. All wonderful, clever people. So the third project is led by Theo Evans from UWA, and his project is called Increasing Tools for the Control of PSHB. So he's got, sort of a number of different aspects to the work that he's doing. The first is looking at biology and ecology of the borer itself, so looking at dispersal behaviour and chemical communication, and that's to kind of assist with things like attractants and repellents as well. The other part that he's doing is looking at those innovative control strategies, particularly chemical and biological control. And he's also looking, as I said, looking at improving attractants for traps, so therefore increasing or improving surveillance and early detection, and also community reporting as well. So, and I think we're all kind of using some nice images that we happen to get of someone in a tree who's doing some important stuff to shot-hole borer infested trees, and I'm glad that's not me up the tree. I'm just going to say that. So, and you'll also kind of see there's an image of some nematodes. So a few of the things that Theo's team is looking at is, biological controls. There are some existing controls that are around that utilise these nematodes, which can actually kill the beetles, and I believe Theo's team is seeing some interesting early results. So another thing that he's doing is using different chemical compounds, including those that penetrate wood, and he will be doing that in field tests with local governments and horticulture industry as well. So he's looking to do that soon, I believe, or he may have already started a bit of pilot work. And this is Theo's project team. So he's actually shown all of his, sort of collaborations as well with some folks at DPIRD, at Murdoch, at Curtin, and a large team at UWA as well. So that's it for me. Thank you.
Now I'd like to introduce the first of our international keynote speakers for today, Professor Shannon Lynch, who's from the University of California, Davis. Shannon is an assistant professor of forest pathology whose research combines large-scale surveillance and predictive modelling to manage emerging tree pests. She co-led California's statewide strategic initiative on invasive shot-hole borers and currently chairs the SISAC subcommittee, making her one of the world's leading authorities on the integrated management of this pest. Many of you may remember Shannon from her earlier visit to Perth, and during that time she conducted field trips to Hyde Park and Lake Claremont and led a DPIRD webinar where she shared lessons from the IPM program at Disneyland. Today, she joins us to share her expertise on how California balanced high-impact tree removals with long-term restoration and the specific research that guided their strategic decisions. Shannon.
Hi, good morning, everybody, and thank you for that very generous introduction. It's wonderful to be here with you all today and to be talking to you about this important issue, and it's an honor to be here. So thank you. So I just want to kind of orient you here on the map. This is a map of California where I am located. This is UC Davis, where I am based at my institution. And so the research I am part of is the problem that I am involved with is basically in Southern California primarily. And the issue is part of that I'd like to acknowledge is, sorry, I'm having issues. There we go, is part of a multi-campus, multi-agency collaboration supporting statewide integrated pest management across conservation, agriculture regulation, academia. And so I just want to acknowledge all of these entities that have been involved and have a shared interest in addressing this problem together. And so recognizing that for my talk today, I just want to first introduce the pest pathogen system in California. It's a little bit different from what you guys are experiencing and kind of put that into a global context. Then I'm going to launch into some of the implications and impacts of host range and how we use that to develop statewide risk assessments. I'm going to explain what we know about management options in California based on what's been tested. And then I'll end with case study in Disneyland, some of the management options that we've learned there, because it's always nice to end with the happiest place on earth. So, what we refer to as Fusarium dieback invasive shot-hole borers. This is a pest pathogen complex. In California, we have 2 of these beetles. So it's caused by these combined effects of these Fusarium fungal species. We have Fusarium euwallacea and Fusarium carotium, and they are vectored by these Euwallacea ambrosia beetles, Euwallacea fornicatus, the Polyphagous Shot-Hole Borer, and Euwallacea carotio, the Carotio shot-hole borer. So these are morphologically nearly indistinguishable, but genetically distinct cousins to one another. So they are functionally the same. And so we like to just simplify them and refer to them as invasive shot-hole borers in California. And so just to kind of take a little bit of a step back, what are ambrosia beetles? Ambrosia beetles are essentially these huge diverse of wood-boring insects that have these special pockets in their body called mycangia. And these mycangia are used to carry fungal spores. And so depending on what group they belong to, they carry these mycangia on different parts of the body. So this is a typical family photo of the invasive shot-hole borer ambrosia beetles within a host tree. And basically they share this ecological strategy with thousands of these other wood-boring insects where the mature females bore into the wood of the host trees, inoculate the gallery wall with the symbiotic fungus that is carried inside of her mycangia, which are located in the mouth. They bore into the trees, but like it was said earlier, they're not consuming the tree. They are solely eating the fungus itself that they depend on for the larvae and the developing beetles itself. So the rest of the family in there are depending on that fungus. The beetle is serving as a vector, carrying the fungal spores in their mycangia and spreading the fungus from tree to tree. This is a simplified life cycle of the pest pathogen complex. And unlike the majority of ambrosia beetles, these beetles attack healthy tissues. So they're going to go for the healthy tissues on a tree rather than dead or dying tissues. So they are going to attack that, and they are going to then bore into the tree. They're going to lay their eggs, feed on the fungus, and then the siblings are going to mate with one another inside of the gallery. That eliminates the risk of needing to look for the opposite sex outside of the tree. And so for that reason, this class of ambrosia beetles does not rely on sex pheromones, which actually has really important management applications. At the same time, then They prefer, once they've kind of mated, the female will emerge out of the tree, and they tend to want to stay on the same tree. So she's going to come out of the tree, she's going to walk around, and she's going to produce another gallery on the tree and stay on that tree as much as possible. It's a really risky business to fly to another location. So they're going to do that as long as possible. And at the same time, the fungus is colonizing onto that tree and causing necrosis and then the tree is also kind of overreacting and clogging up the xylem vessels to try to prevent the fungus from moving systemically through the plant. That chokes the plant as well. So it's a combination of the fungus colonizing and the choking that causes this dieback and sometimes tree death, depending on the tree, depending on the location and the context. And then when the fungus when the host can no longer support the production and the development of the fungus itself, that's usually when the beetle is going to flee and go to another tree. Oh, okay. So this story becomes a little bit more interesting because it's not just Fusarium that these beetles carry. First, they carry these other species, including a Graphium species and a Paraacromonium species. They play an important role in the life cycle of the beetles, but the Fusarium species are the dominant ones in the mycangia, but they do have this kind of community of other fungi in their mycangia. This is a global distribution of all the Euwallacea species where they have been introduced around the world. This is the native range. So Euwallacea, there's a whole range of Euwallacea beetle species. They are native to Southeast Asia. Not all of them are destructive or attack living healthy trees like PSHB or the KSHB, but all Euwallacea species have carry the Ambrosia fusarium species, which are part of what's called this Ambrosia fusarium clade. So it's a special class of fusarium species that they carry. This is the global distribution of PSHB, where it's currently in Israel, California, South Africa, Western Australia, as well as Spain now. And we used to think that this relationship between these Fusarium species and the beetle was, this one-to-one relationship where, for example, PSHB only carried Fusarium euwallacea, but we do know that that relationship is not exclusive now because you guys have this Fusarium species AF18. So these were all introduced around the world separately and separate introductions, but we happen to have in Israel, California, South Africa, as well as Argentina and Spain, we all get Fusarium euwallacea, but you guys get the special fungus, so you guys are special. So coming back to California, so this is a map of the distribution of PSHB, KSHB, and where we find both beetles in the same county. PSHB was first detected in California in 2003 in a state monitoring trap in Los Angeles. At the time, this was PSHB was not known or understood, and so we thought it was the Shot-Hole Borer. So the problem wasn't recognized until 2012 when it was discovered on a backyard avocado tree in Los Angeles. And then the second introduction, the KSHB, was identified late in 2013, where it was infesting commercial avocado groves in San Diego County. And then in November 2023, nearly a decade after its northernmost detection, PSHB was then confirmed in infested sycamores in San Jose. So this is about 450 kilometers north of the previous leading edge of the infestation, raising major concern about its spread now in Northern California. And then, just to make things even more interesting, in October of 2024, a separate infestation in Santa Cruz County was discovered on box elder. However, the California Department of Food and Agriculture pest detection labs confirmed that this infestation was a new introduction of a third shot-hole borer beetle species. This is Uelacea interjectus, which cultivates Fusarium floridanum. And so this beetle is much larger than PSHB. So this is looking at the two beetles on a penny. The Uelacea interjectus is on the left. We like to refer to this one as like one on steroids, or it looks like the Hulk compared to PSHB. It's a lot bigger, but it has that Fusarium floridanum, the conidia, characteristic of those Ambrosia fusarium species. They have these dolphin-shaped conidia. Also has in its mycangia this Paraacromonium species as well. Uelacea interjectus was introduced from Southeast Asia to the United States on 3 separate occasions in the 80s. And it's been pretty much just kind of in the Southeast United States. So this is the first introduction in Western North America that we found so far. It's been detrimental to populous species in Argentina and China, and has affected Japanese fig orchards in Japan. But so right now we're just really trying to get our arms wrapped around this and understand what's going on. So it's pretty much just in this region. It was introduced next to this woodlot here near this state park at the mouth of this riparian corridor called the San Lorenzo River. It's moved down within a year into, this is Santa Cruz, the city of Santa Cruz. And so now it's moved into the city of Santa Cruz through this riparian corridor. And so there is concern about what's going to be happening now that it's kind of moving into the city. It's mostly moving through box elder. So it's just kind of ripping through. The symptoms are the same. Everything looks functionally the same. It is also attacking other tree species, but we're just seeing hits here and there. A little bit more hits on our poplar trees, our native poplar trees. The interesting thing is with box elder, this is actually native, part of our native vegetation in Northern California. So these are not street trees. This is a dominant part of our riparian habitat. So it is actually an important part of our ecosystem in this area. And so it is a concern that it's going to get lost. So right now, what we're trying to do is understand what the potential host range of this beetle is. We're doing trap optimization studies and setting up monitoring plots to just get our head wrapped around it. So for the rest of my talk, I just wanted to let you know about the third beetle species that we're dealing with. But for the rest of my talk, I'm going to be focusing on the other 2 beetle species, because that is what we have our historical data on and our understanding of much more about the spread and everything. So as far as the host range is concerned, very early on, we observed a whole range of symptoms and a variety of tree species. This is just kind of just some examples that you would see and the kind of response that the host produces depending on which species gets attacked. And because we observed a lot of variation in the severity of the impact, where some hosts just show branch dieback when they're attacked, and then others get killed. We wanted to understand this variation across species. And so we dug into the biology, and we found that when the beetle tries to attack a tree, it could not successfully colonize on some of those trees. It would just try to produce a gallery and not successfully colonize. While for others, the beetle could not establish a gallery. The fungus could colonize, but it couldn't establish a gallery. The fungus can't spread from tree to tree, but the fungus could colonize on the tree. And then for other tree species, a beetle could bore into the trunk, produce a gallery, establish a fungal garden to feed on, and then make their brood and spread onto a new host. So both the beetle and the fungus must be able to reproduce for a host to be what's called competent. And each of those steps have the potential to differ in susceptibility among species. And fortuitously, some of the most heavily infested areas in the very early stages of the infestation in Southern California are home to two of the biggest botanical gardens. And because they harbor a wide range of plant species that are unique and represent common ecosystems from all over the world, including those that are native to Southern California, we had this rare opportunity to use those gardens as this basically natural laboratory to understand host range of the beetle fungus complex. And what we found was that these host types are based on these nested outcomes between interactions between the host plants, the beetles, and the pathogens, where the beetles make an attempted attack on 324 species, 137 of which only the fungus can colonize. A subset of 77 species are competent hosts that support reproduction of the fungus and the beetle, and are important in the spread. And that includes 20 that are native to California. It includes avocado, and anywhere from 25 to 60% of all individuals that are planted in the urban landscape in Southern California. But then a subset of 18 species can be killed when they are attacked. So the lethality of different hosts is important in terms of ecological impact. So these hosts are distributed over a very complex landscape, including native plant communities, urban forests, as well as avocado groves. And depending on the host, depending on the location, depending on the context of the environment, the impacts of these beetles, they can be devastating. So this is an example of a riparian forest in San Diego County, just right along the Mexican border, and where within 1 year, over 200,000 willow trees were killed when the beetle infested into that region. So this is kind of the worst case scenario that we have witnessed when this happened, and there were cascading effects of this mortality. So this is looking on the ground of that mortality within 1 year. And then 6 months after that, we had castor oil come in and encroach into the region, this highly invasive weedy plant, preventing recruitment of our native willow species that serve as critical breeding habitat for our endangered bird species. And then in the urban environment, severely damaged trees were removed. Thousands of trees have been removed in Southern California that pose high safety risks from fallen limbs, threatening millions of dollars in damage and replacement costs of nearly $16 billion. Removal costs are really high for these trees, up to $1,000 per tree. And the loss of ecosystem services is also great. They've valued these at, around $987 million annually. The story for avocado is kind of interesting. So once established, the beetle does spread very rapidly within groves and across regions. So for example, in Ventura County, this was a newly infested region and a major avocado growing area in the state, where we had 3 groves that just got infested, and then within 1 year in our survey, 10 additional groves were infested. In San Diego County, where infestations were more established, the prevalence ranged from 20 to 75% in groves, and then increases went up to 35% within a grove in a single year. So it can move quite rapidly. However, This is just the California experience. The impacts on avocado industry were less severe than initially anticipated. This is largely because the beetle preferentially attacks the crown rather than the trunk. And so it establishes those galleries and reproduces in the small tertiary branches that are roughly about 11 centimeters in diameter. And so because of this, because the beetle rarely establishes on the trunk, it doesn't kill the tree. And so because pruning is already a standard practice in avocado production, it also has served as an effective tool for managing PSHB by removing these infested tertiary branches. And that reduces the beetle populations and propagular pressure while also maintaining tree vigor. And so that has helped. The problem where the greatest damage occurs in these groves is where the sanitation is not maintained, especially in areas where there is Botrysphaeria canker and other canker pathogens. So first, these holes serve as infection courts for these canker pathogens to enter. And so that provides pathways for these canker pathogens to spread. And the beetles themselves then carry the fungus on the surface of their body and then kind of exacerbate the problem as well. And then that actually creates a lot more branch dieback. We also see on the trunk, if you then get a lot of propagular pressure on the trunk without pruning the branches on the side, if you get a lot of propagular pressure on those tertiary branches, then it can spill over onto the trunk eventually. And then you get establishment of those canker pathogens, and then that's where you start to see a lot of the dieback and things like that. So poor sanitation without active management, that's where we see a lot of problems occur. So because this is a multi-land use and stakeholder issue, we realized that we needed to develop a framework that addresses the complexity of this issue so that we could take action using whatever knowledge is available at any given time. So our approach was to develop an IPM strategy as part of our research program. And the essential IPM components that we identified were monitoring and early detection tools, developing those tools to identify those disease-causing organisms and detect new infestations, researching their basic biology and life history, conducting risk assessments to identify which tree populations and forest communities are most vulnerable in establishment and most important in their spread, developing and evaluating preventative and curative biological, mechanical, and chemical control options that are tailored to the appropriate settings, and then sharing the menu of options and improving IPM strategies with stakeholders in an iterative process. And so something just to emphasize is that none of this is a linear process. It's not something that you check off these boxes and then you do, and then you're done. This is something that we're constantly, continually refining and iterating as we learn more information and get feedback from stakeholders as well. As far as the risk assessment's concerned, to do this, we have established a large plot network in infested and non-infested locations throughout the state, from San Diego to Ventura County. So this is just the southern part of the state. These are in locations in peri-urban wildland forests, as well as avocado groves, incorporating the whole range of conditions over which the beetle and the fungus are able to thrive. We've collected microclimate data in each of those plots and measured the amount of disease, if present, on each tree species. And we've incorporated host composition information. And because we've measured microclimate in each of those plots, we can calculate the number of beetle generations that can occur in each of those plots. And we developed the model and we validated the model and basically used that information from the model to try and predict where the beetle is most likely to go and establish in the state of California. And this is essentially what the map looks like when you combine community composition and this bioclimatic map for beetle generations. So the host composition is these clusters. You can see them more closely in these zoomed in boxes, but it's these clusters from yellow to dark red. And those represent locations where you have tree composition, areas where you have trees that are actually more closely related to one another and combined with their abundance. So the more trees you have that are more closely related to each other are actually going to be, those locations are going to be more susceptible, more likely to have an infestation. And they're actually, you're more likely going to see those areas where trees are going to be killed because the model incorporates those killed competent hosts in there. So you're going to see more severe impacts in those locations as well. And then also we have the microclimate impacts on the beetles as well. So the light green to dark blue, the dark blue areas are going to be more severely impacted. And so this is kind of basically showing that what we found in our models was that the temperature kind of mediates the effects of host composition. Host composition is the primary predictor of the likelihood of beetle establishment. But in areas where the community, the tree community is like less favorable, if you have a good microclimate, then you will have beetle established there because it's favorable for, you can get more beetle generations. So warmer areas with more beetle generations can support establishment. The other thing is that this is, for example, San Jose area where we have the beetle that has established. This is Sacramento area where the beetle has not established. The host distribution here has a lot more hot spots and the temperature is much more favorable. So we're really concerned that given much more favorable compared to San Jose area. So we're much more concerned that the if it establishes based on the host, that it's actually going to even spread more quickly because the temperature is more favorable. So that's how we're using the map. We've also converted this into a interactive tool for stakeholders to use on the ground. They can use this on their smartphones to basically identify locations where to monitor, where to apply treatments, where to target tree removal efforts and other control options. So the model is basically used as a tool to assess risk and guide response priorities, like identifying locations where control treatments will be most effective, help reduce the use of pesticides, and inform to, where to strategically remove trees, reduce costs, and also where to monitor. So all of that risk modeling is to basically help us decide where to apply the management options that have been tested and what we know about in California. As far as monitoring concerns are, visual and trapping are the tools in our toolbox. They are not mutually exclusive. It's not one or the other. It's using both in combination to as our tools in our toolbox. So monitoring, again, happy to share these slides. We have a great YouTube video for trapping, on how to place traps, where to place traps. We have two kinds of traps, lingering funnel traps or sticky panel traps. For our statewide trapping efforts, we use the sticky panel traps because they're more cost effective. They're just easier to switch in and out. And We use the Lure Corciverol because that has been the most effective in all of our tests that we have ran in our trials. And the uses of these traps are for early detection, guiding inspection efforts, and for finding the best timing for treatments. The lure itself is not attractive enough for beetle control. So this is not a tool, again, for baiting the beetle. It is a tool for making decisions, and that's really important. And then one PSA that I have to mention, because we've learned our lesson here too, is not to hang traps on a branch or on a tree or put them under the canopy or near a tree, because when you do that, you're effectively treating the tree as a bait. And you will basically bring the beetle to the tree and it will attack the tree. We've done it. We learned our lesson. So I'd just like to impart that lessons onto everybody. So this is just highlighting and showing, the importance of how trapping can be used on a local level. So this is 2 graphs just showing when within the same county, the trapping patterns. So trapping or the beetle activity is typically twice in a year. They usually peak in the spring and in the fall, but there is variation among sites. And so that's why it's really useful to use trapping on a local level to say, okay, this is when I'm going to apply my particular treatments or my particular decisions at the local scale. That is why the trapping is so effective. And then for visual inspections, what we're looking for is signs and symptoms. So signs can include anything from looking for the beetle itself. So the female likes to guard her entry hole. She'll stick her little abdomen out and guard her entry hole there. So that is a nice sign of the beetle. And then depending on the host, you'll see different responses. So looking for that staining or frass, or for the avocado, it likes to dramatically produce the sugary volcano. It's that parasitol exudate, or some trees produce gumming. But these symptoms are always associated with that perfectly round entry hole that is the size of a tip of a ballpoint pen, roughly 0.85 millimeters in diameter. And so if you kind of remove that staining or gumming and you look for that perfectly round shape entry hole, then you're close to that beetle. Visual inspections, just kind of having tips and tricks, knowing which side of the tree the beetle tends to attack first, just helps in your efficiency of monitoring. In Southern California, it tends to be the north side of the tree. In Northern California, it tends to be the south side of the tree. They like the side of the tree that's warm. So it's going to differ depending on where you are. So just kind of orient and get your search image there, but always inspect the entire tree, but know where to inspect first. Look everywhere, because they will attack the branches and the trunk as well. So have your binoculars. I think it's really important to assign an infestation level, because infestation level is going to dictate what actions you are going to take from there. So low infestation level is less than 50 holes. Moderately infested is 50 to 150. Heavily is more than 150. And then severely infested trees have more than 150 holes and are exhibiting severe dieback. And then activity is also really important. You can have thousands of holes, but if the beetle is not active in that tree, then there's nothing going on in that tree anymore. So beetle activity is also going to determine how the tree is responding. Some cases, if you see this callousing, this compartmentalization, the tree actually might be recovering and effectively walling off the beetle and pushing the beetle out. Or the beetle might not be there anymore, or the tree might be dead, or the beetle just might not be there anymore. And so it may not be worth treating the tree. So it's just really important to know the level of activity that is on there, because that might dictate whether it's important or not to remove a tree. You can use latex paint to determine activity of those entry holes as well. So the beetle, their entry hole covered, they chew through everything, just so you know. So they will chew through anything. So if you cover that hole, we take advantage of that behavior. We use this white water-based latex paint, cover up the hole, and if you find that the hole is open, then you know that there is a beetle colony inside of that entry hole, and that there's an active family in there. And that just helps to confirm whether the the treatment that you're applying worked or not. And so as far as integrated pest management control options, we have 3 kind of tools in our arsenal. There's cultural, physical, mechanical, there's chemical options, and there's biocontrol options. For cultural, physical, and mechanical, this is the most important tools in our toolbox that we use. Includes pruning infested branches, removing amplifier trees, so the trees that are like really pumping beetles out, correctly disposing infested material, and restricting firewood movement. When it comes to removing those amplifier trees, what we're talking about is these severely infested trees. In California, that's what we're prioritizing. These are trees that have more than 150 active entry holes and are exhibiting dieback. These are the trees that are big sources of beetles. These trees have a compromised vascular system, so the treatments, any treatment that you try to apply is not going to work. The system is too compromised, and they are hazards. And so the tree is just too far gone, and they are going to be your source of inoculum. So this is what we want to remove to reduce propagule pressure in the area. When the trees are removed, we also find that it's important to remove the stumps, especially because if there is moisture in the stump, they can support the fungal growth, and they can be sources of beetles and continue to pump beetles out. At the same time, if you cut them and they can no longer support beetles or support the reproduction of the fungus, what happens is, if you have something where you have all of these beetles and then all of a sudden they have nowhere to go, you create a mass dispersal event, and they're just going to rain beetles onto the next tree and inoculate that. And so it's really important to remove the stump with a stump grinder. Apart from that, then it's really a matter of reducing inoculum by removing branches. And so removing infested branches and correctly treating those branches is going to be really important to reduce propagule pressure. This, for our oak species and for our avocados, this is especially a good option. But for most of our trees, this is what we typically do to reduce that propagule pressure. And we don't dress the pruning wounds, but we will spray the pruning wounds with the Bacillus subtilis, which is produces these antibacterial biotics that prevents secondary pathogens from entering into the pruning wounds. And then we correctly dispose of the plant material so that we're not spreading the beetles around. We do this because this is depending on the host. Even those that can be killed when they are attacked, they can recover. Sometimes we do see recovery if they are not getting bombarded with beetles. And so active management, active pruning, active removal of these amplifier trees reduces the beetle pressure. And then it gives those trees a fighting chance to compartmentalize and prevent and push the beetles out. And so it's really this combination of monitoring with traps and visual inspections and applying these cultural and sanitation practices to make sure that these trees are recovering and keeping an eye on things, and removing branches as needed is to keep the population at bay is the important thing that we find to be most effective. So as far as green waste and firewood, these are pathways for spread. ISHB typically only spreads to neighboring trees. Again, so the biggest way that it's going to spread long distances is by people. So the joke here is that these beetles are not good flyers, but they can spread up to 70 miles per hour, like on the back of a truck. So management of infested plant material is really key to preventing its spread. Chipping to, our studies show that chipping to less than 2.54 centimeters or as small as possible kills 99% of the beetles. You can use the chips locally as mulch on the same property. If you chip and compost, you have pest-free mulch. If you can't chip, you can solarize the logs with a clear tarp, or they can be kiln dried. The fungus is not airborne, so it's not going to spread from tree to tree. So it's really about killing and making sure that you're killing the beetles. So just please don't move firewood. So biggest thing is to focus on the cultural and mechanical control. As far as chemical control, this is a tool in the tool belt, in the toolbox. There's trunk sprays, systemic soil injection, and drench, and there's trunk injections, but there's some important considerations that need to be conveyed. So the first is that only consider treating infested high-value trees. There's no preventative treatments when it comes to chemical treatments. This is not a preventative option. Generally reserved, I'm not even going to say for heavily infested trees, it's really a treatment that is for moderately infested trees that are high value. So it's a very small subset of trees that we're talking about that this is maybe even an option for. Heavily infested trees, the vascular system is too compromised. The pesticides are not going to be moving through the wood. The most effective that we've seen in our trials is a combination of insecticide and fungicide, but you need to consider, again, the tree health, the location, the value, the level of infestation before applying these treatments, because that context is going to really dictate whether or not it's going to be something that you can use. Pesticides that are being applied need to be done by somebody who knows what they're doing, by a state licensed person. And like, I should also say that these are the considerations in California, based on what we have tested, and none of these treatments work 100%. So the biggest message, this is not a magic bullet, not preventative, not curative. It is a tool in the toolbox to help keep things at bay. So we have trunk sprays that are appropriate on heavily infested trees. that can be done or can be applied locally after you remove a branch. And the goal is to kill beetles that are about to disperse, that landed on the tree, that might be looking for another host. So these should be applied when the beetles are dispersing. And it's important to avoid flowering plants and areas close to water. The systemic pesticides, these are pesticides taken up through the tree. So killing the insects and fungi in the wood, this depends on tree uptake. They can be applied in the spring or the fall. The trees need to be irrigated before and after treatment. And this is why these things aren't the best, they're not preventative or curative. So we've got imidacloprid, which is a soil injection. It is good for sap-sucking insects, but PSHB is not a sap sucker, but this happens to be more systemic than the trunk injection emamectin, which is more effective in lab trials. But emamectin is not as systemic through the wood as imidacloprid. So that's where the trade-offs are. And so that's where we have, that's why these things aren't as, our magic bullets, if that makes sense. Problem with doing the injections is then you have the risk of opening up wounds and creating infection courts for canker pathogens to come in and cause actually more damage. And so these can actually be more damaging because they can, fusarium grows pretty slowly compared to like a lot of these canker pathogens. And so if you do these injections, the canker pathogens can actually grow more rapidly through the phloem and girdle the tree and cause more damage. So these things need to also be applied over and over and over again. So it's again it's not something that you want to do on every tree. So even once you get to the last resort of needing to apply a chemical, the decision on what treatment to use really does still depend on the location. If you're next to water or whatever, because and it's tree health. So the main message is that you shouldn't be applying pesticides just because you have an infested tree. The goal is to apply other tools in the toolbox first. So just to reiterate, there's no preventative treatments. This is generally reserved for moderately infested trees that are high value trees. Must be a licensed applied pesticide applicator to apply it, and none of these treatments work 100%. And just to kind of hit this home even more, we're applying these things depending on the context. So we're taking into consideration the host type, the infestation level, the tree hazard level, the tree value, and all of those factors go into this matrix of like what activities you're going to do depending on those different factors. And so it's really important, and all of these involve monitoring. Whatever actions get taken, monitoring is always the follow-up, or it is always the guide. So monitoring, and then usually it's those cultural practices. Okay, the biological, I don't have time to get into that, but it's an active area of research that we are doing. And so it's something with natural enemies, entomopathogenic fungi, and endophytic bacteria that is ongoing that we don't have results yet that are really viable. And so one thing I just want to end with is that in Disneyland Resort, just ending with this case study of how we've applied these tools and tactics in Disneyland. This resort has approximately 16,000 trees, 681 tree species in the area. And so it's a hyper diverse place. that is an amazing urban forest that you wouldn't think of because you just think of rides and Mickey Mouse. But as a response, they had infestation of PSHB. And as a response to that infestation, Disneyland injected preemptively 600 non-infested trees in their park with systemic pesticides. And so bless them, they wanted to do the great thing, but what they ended up doing was damaging a lot of their trees and causing this swath of canker pathogens throughout the park and causing a lot of damage. And so we came together and developed an IPM program for their park where we implemented traps around the perimeter and throughout the park itself to monitor hotspots of beetle activity. Once we identified those hotspots, we monitored inventory trees to determine the source of the beetle populations of infested trees and determine which management actions to apply based on attack severity and host type. And so we did monthly visits to evaluate the findings. We applied the cultural sanitation and chemical treatments, but we reduced the amount of chemical treatments by 90% of the park just by implementing these cultural practices alone. And we followed up with trainings and workshops. This is a really good example of just that early detection. Because of all of the monitoring, we would catch something early, remove the branch, follow up by monitoring, seeing that there was still attacks going on. So they treated this further with contact insecticide bifenthrin. And then that allowed the tree to recover and gave that tree a fighting chance, and it's been able to heal. And so just to kind of reiterate, the fact is we use active monitoring, we established guidelines when management was needed, we applied a combination of cultural and chemical tools, and we used training and outreach, and we rinsed and repeated, and we reduced the beetle population in the park. We didn't eradicate it, but it got to a level that we could tolerate the population in the park, that the trees in the park are able to recover and not get completely bombarded and die back. So I just want to say that and end with this, that the beetle in California is here to stay, but with active management and timely action and monitoring and an investment in resources, it can be managed if people educate themselves about the beetle, if they take the preventative action and avoid problems and use science-based management options. Use the tools in the toolbox and leave the chemical management as a last resort. And so with that, I just want to say thank you and looking forward to having more conversations.
And Shannon, I know there's some questions in Mentimeter for you for later, and if there's not, I've got some anyway. So next, I'd like to introduce Professor Francois Roets. He's from the Stellenbosch University in South Africa, and after completing his PhD in 2006, Francois held a postdoctoral fellowship at the University of Pretoria before joining Stellenbosch, where he currently serves as Professor and Chair of the Department of Conservation, Ecology and Entomology. He is a global authority on multi-organism interactions. His multidisciplinary research repertoire covers diverse fields, including landscape ecology, molecular phylogenetics, genomics, and metagenomics, with approximately 175 papers in peer-reviewed journals and numerous popular articles. Francois has been instrumental in tracking PSHB infestation across South Africa, from botanical gardens to commercial orchards. Beyond his research, he has mentored more than 100 researchers, including 18 PhD students, 7 postdoctoral fellows, helping to shape the next generation of biosecurity experts. He will share his unique perspective on how the pest adapted to host species across different climates and the lessons learnt in coordinating the national response. Thank you.
Thank you very much for that mouthful.
Really well.
Yeah, thanks for inviting me and letting me see what you guys are up to. I have to start off by saying that South Africa is a good example of how not to do it. We don't have the resources, and generally people only care about this cute little beetle once it's already infested something and starts killing something, and yeah, by then it's too late. So I'm from the Department of Conservation, Ecology and Entomology. So our department, on the one side, we're trying to save the things that are very few of, and on the other side, we're trying to get rid of things that are too many of, like this little beetle. And this presentation always sort of leads towards, can we stop it? Now in South Africa, that bullet has left. In Australia as well, stopping it is going to be extremely difficult. Containing it is going to be probably the best option. Right, so in South Africa, we have a very similar experience to California in terms of the management options, so I'm not going to go into too much detail into that, because that's Shannon dealt with that quite in detail. I'm going to stop and just look at why this beetle is so invasive. And the first thing there is it is really, really small, so which means you don't actually notice it. It's very difficult to see. And in South Africa and you in Australia as well, there's many, many, many beetle species that look the same. So you really have to spend time and get an expert to identify this. They also hide in wood, living wood, but also in dead wood that's still fairly fresh. Around about a month or so after that wood has been severed from the tree, the beetles will still be alive in there and can still actively disperse. Which means it can be transported with untreated goods, untreated wood, even in infested plant material, living plant material. And in South Africa, we have this sort of big market of little trinkets and things that people make out of wood that people can buy, take home. And very often that's made in infested areas of actually infested wood. So when you buy a wooden figurine in Africa, put it in the freezer for a week, and then it should be safe. So the initial symptoms also on living trees are obscure. So probably one of the easiest trees to spot it in is a picture there is a plane tree. You can see a nice dark stain on that. Most other tree species is really difficult to see if there's only one or two infestation points. So you need to train yourself to be able to spot this beetle and also to know what to look for and what different tree species. Right, so invasive. The biggest issue here is that they have incredibly fast generation times. If you look at the numbers there, a single female can lay 30-odd eggs, and the adults can reach, depending on the temperature, within a month. You'll have a whole bunch of adults that can disperse from there. They have this weird reproductive strategy. It's called a haplodiploid mating system, which means that the females can produce males when they want them. So if a female has left the nest and hasn't mated yet, so you can start a new gallery system on a host, lay those eggs, and then mate with their own siblings, with their own sons, in order to get that reproductive cycle going again. They also have these multiple overlapping generations. If you look at that gallery system there, there's the moms and the sisters and the brothers in there, but also some of the babies, the larvae in there. So they continuously produce beetles. It's not just this sort of once a month pulse that happens, it's continuous that happens, which means that they build up numbers really, really quickly in susceptible hosts. And if you do the calculations for this, if every female survives, it really is this sort of logarithmic increase in numbers in warm times. Another reason for its invasiveness, which you also experience here, is the multiple of hosts that they can infest. So, I mean, these host lists should always be taken with precautions, because it depends on where the host is found, under what conditions, the condition of the host itself, all kinds of things. So I just updated the list for South Africa 2 weeks ago. So we're currently up to 181 recorded host species. There's no active monitoring going out throughout, or statewide, nationwide monitoring happening. So we actually don't know how many hosts there are. Of that, we have now 91 of our species that are on this list, of which about 50 is reproductive hosts. So the beetle can reproduce in them, and some of these and many of these tree species are actually in deep trouble. But this is still being updated as we go on. A little bit of the history, I think this has been mentioned. So it's from South Asia or Southeast Asia originally. So Israel and California played with it for the first time. That's a long time ago already. So you've been struggling with it for a long time. Hawaii, it's been reported in 2007, but I think it's been there for longer than that. South Africa, we were next in line. So it was officially reported in 2017. Sorry, that's the date of the publication, 2018. But we have a sequence of this beetle in the country close to one of our ports from 2012 already. So it's been in South Africa also for a long, long time before people actually started noticing it. After that, it was Italy, then you guys, South America. It's quite widespread in South America now. So South Africa used to have the biggest continuous outbreak of the beetle in the world. I suspect that South America, somewhere in South America, that's now. And most recently, Spain. So this is just where the beetle has been detected outside of quarantine areas and outside of greenhouses and things like that. So where they're actually in the outdoors. So how does it get around? So shipping is the most obvious answer for that, to get around. And it's probably not so easy with live plant material because this gets inspected quite easily, but rather through untreated wood, like wood pellets. So everybody inspects what's inside of these crates, but not what's carrying the crates around. Also the ballast wood, the weight sort of adjustment in these ships are often very sort of untreated, bad quality wood that you can't use for anything else. So there's a picture of one of the planks at Richards Bay Harbour in South Africa, lying on sort of just downloaded from a ship that came from who knows where. And inside that piece of wood, there were living beetles there, living fungi. Origin we don't know, and species we don't know. And this continuously happens in South Africa, and I suspect also around the world. So in South Africa, this is just a genetic tree of some of the beetles or PSHBs around the world. And those two sort of highlighted areas are the two different genetic varieties of the beetle that we have. And we now know that the beetle has been introduced in the country at least twice. Once from somewhere in China and once from probably in Vietnam somewhere, which also introduces more genetic diversity for the beetle and the fungus, which means that they can recombine and start sort of evolving towards conditions in the area. If you only have one clone, they can't actually adapt over time for new conditions. Once in the country, that's where the real trouble starts and how they move around. So in South Africa, there's a whole massive industries linked to wood. We like to make fires, we like to braai. Tree removal companies move this around quite a bit. Also the firewood industry. The problem in South Africa is we cannot actually close borders, provincial borders or anything like that for this. We'll cut off the livelihood of thousands of people in the country. They tried it and it's not possible in South Africa. Nursery trade. So these are some plants from close to Joburg and Sandton area. In a nursery there, just walking around there. The beetles don't establish in all of these, but in one or two of these plants, if you actually open it up, they form these sort of circular gallery systems in there, sort of rotating towards the middle, they spiral form, and they actually form viable colonies inside anything that's a host from 2.5 centimeters in diameter and up. So really small plant or thin plant material they can also infest. Wood chips can be a form of this beetle moving around, especially if it's very coarse, like Shannon mentioned. So we need to chip it very, very small, and then preferably don't move it from that site. There will still be a few beetles that's alive in there. And what we know from the life cycle now, you need one beetle to find a host tree in a new area, and you can potentially have a new outbreak. So some of the examples from South Africa that we could trace. So this is a pile of wood that was found in Lanseria, all the way from Sandton, and this was actually used at a brewery. So they boil water or whatever to make beer, and they got one of those tree felling companies or wood dispersing companies to get some wood there. They saw some holes in it. So there's a piece of stump that was sent to our colleagues. You can see all the little bit of holes in there. Just putting that into a dryer to chase out the living creatures in there. 44 mature females were found and 5 males. Inside that wood, there would be still many, many larvae and eggs also in there. So that piece of wood is about 500 grams. So it's like a bucket of margarine, basically. So if all of those guys, all of those females found a host, you'll have about 1,408 baby beetles within a month, and this will continue. And as it happened, that pile of wood was dumped right next to one of his preferred hosts in South Africa, an oak tree. So when we went back there, about a month later, there was already some dieback in one of the branches of that tree. So that was the origin of one of the outbreaks there in that area. As Shannon mentioned, things can happen really quickly. So this is again in Johannesburg, just outside of Johannesburg. So this is what's nice about Google is you can go back in time, the images there. So it's a street view map in 2013, and then 2018, all of the trees were dead and gone. And at that stage, nobody knew what was happening to the trees, but we could actually look at the stumps, what was left in the area, and we could find the PSHB and the fungus still thriving in those stumps that were not treated and were not taken out months later. Just another example there. So this is a plane tree like you guys have everywhere here. Unlike the plane trees around here, PSHB seems to favor starting in the base of the trees and then work their way up in South Africa. But again, so that tree, if you look at, that's one of the branches there, within 2 years, it looks like somebody took a shotgun to that tree. So it can happen really, really quickly. A little bit closer to my home. So there's a map. It's close to Cape Town. So Somerset West is part of Cape Town. Cape Town. That's where I live. I did not bring the beetle there. So this is just basically around about COVID time. So we know the exact tree individual where this infestation started. And the reason for that is it was a lady that redid her garden, got some people to help her with that, and they imported some logs all the way from an infested area, oak logs, to do terracing in the garden. And right in the center of the garden, there was a box elder tree. And that was, as you guys know, is the favorite food of this beetle. So that happened in 2019. We had this one tree infested, and we started monitoring this. Unaided, a year later, it spreads about, in these conditions, about 3 kilometers a year. So half of the Somerset West area was infested. And at that stage, we realized there's going to be a problem here because there's no green waste facilities anywhere nearby. The closest one is in Stellenbosch, where the university is. There's a green waste facility there. So we actually started monitoring up the highway there. And lo and behold, you can actually track the movement as people take the infested wood to the green waste facility there. And 2 years later, we found it in Stellenbosch, and it's literally infested thousands of trees there, which are starting to die now. At the same time, it also jumped about 40 or 50 kilometers to Cape Town central area now, Stellenbosch National Botanical Gardens, those areas where it's also infesting. And the impacts can be very severe. So this is in the Knysna area, George Knysna, sort of the more wetter areas in South Africa. These are all oak trees, just 3 trees in 3 streets, just one day taking the pictures there. These trees should have bright green canopies. And this, as I said, is an example of what not to do. If a tree looks like that, it should have been removed months ago already. These things are pumping millions of beetles into the surrounding areas and right next door to some of our most iconic native forests. I'll sort of mention a bit about that. Lots of trees of cultural significance. So there's around about 200-year-old oak tree in front of a very old church there. Rich history of this tree. I mean, they diverted the roads, opened up the spaces around it to make this tree happy and healthy a couple of years before that. All kinds of signs everywhere. Looking at a closer inspection of the tree, and you'll probably see similar things happening in Australia now where people do some weird things. Everybody has a cure. So somebody in the municipality there decided they're going to put the sort of a drip system on the tree. So those little buckets you see there's kind of like a drip when you go to the hospital. All kinds of chemicals of who knows what in there, with little pipes going into the tree. If you look at the size of the tree versus what's actually going into that tree, there's nothing happening, and it's too late. So there's that same tree. Again, don't wait until that happens. You go to those branches, there are millions of beetles on a warm day flying, literally there's clouds of these beetles that can fly around. If you do not reduce that propagal pressure, they go for almost anything in the surrounding areas. So then the tree was chopped down, and that stump was actually turned into nice tabletops for the church. Quite pretty, actually, because you can actually see the beetle lines, those black lines scribbled in there. So you can treat the wood and actually use the wood afterwards. I think they steam the wood at a high temperature to kill the beetles. So at least there's some remnant of that tree somewhere. It's not a very clear map. So this is the situation in South Africa. So all the dots that you see there is where we've confirmed with DNA evidence, beetle or the fungus. These blue ones are the more recent ones since COVID. As I mentioned, nobody is monitoring in the country. So we're literally getting samples from random people, interested people, so we can sort of pinpoint where it's from. It likely started there on the East Coast, at Durban Harbour, Richards Bay Harbour, where the beetles came in for the first time. And you can see these huge gaps between these clusters in the distribution there. That's where humans have actually transported these beetles, and that's the sort of centers from where they have spread. So everything in that sort of blue area is from 2019 up till now, where we found the spread there. As I mentioned, no monitoring. Yeah, so I'm really bad at dealing with my own finances. So we teamed up with some economists just to have an initial idea of what this is going to cost the country. See, these models are probably way off because at that stage we didn't have a lot of information. It's probably going to be higher than expected and not lower than expected, even though there's some of the management going on in South Africa because of the resource. Well, there's not a lot of resources available. These trees are left there. The rate of spread is actually increasing from what we see. And most of the impact, this is in millions of US dollars, will be on urban forests. So that's the top one there that's encircled. Forestry, so that's our native forest. That's a severe underestimation because at that stage we did not know the host range. Wattle trees, acacia trees, they do like them under certain conditions. We have wattle forestry areas. Avocado trees, it's managed, it can be managed on avo trees, like Shannon mentioned. It's not in our avocado growing areas currently, so we actually don't know what the South African impact would be in those regions. But yeah, so we're learning a lot more. We probably have to revisit all these calculations now with the new information we have, and also sort of the rate at which certain plants die. For us, and from my sort of research perspective, I'm really interested in what's happening in the natural environment. So in South Africa, we have about 18 different forest types. And fairly early on, we set up permanent monitoring plots in 7 of these forest types. With help of colleagues in Pretoria and Grahamstown. There's a MSc and a PhD student that was looking at these monitoring plots, revisiting these monitoring plots every year. I think we're up to 5 or 6 years now, and literally counting holes and seeing how this progresses, looking for tree death. And some of the initial sort of results there, we can see that there's about 10% of the tree species, of our native tree species in this forest are susceptible. We find it in all the forest types, except for the mangroves. So all of our forest types that we looked at are in some sort of danger or will be impacted by the beetle. What is very clear is that you also find it in these sort of picnic sites or these areas where people visit in the middle of pristine forests, far away from the front of the movement of the beetles. So there's an example of our national tree, that stump there, you can see little toothpicks sticking out of it. So I just stuck those in there so that you can see where the holes are. So that's a gymnosperm, and it's a breeding host for PSHB. To the sort of, to the right of that, you can see some pieces of wood lying there. That's remnants of firewood that somebody brought to the place. There's a fireplace right next door, and these beetles just jumped into the nearest tree in that forest. So this is the place where we stayed one weekend while doing our surveys, and in that bag of wood, the guy had infested wood with PSHB also. So we often have these slogans, buy the wood or burn the wood where you buy it. In this case, it's not a good idea. They bought the wood in George. That is still in George in the places there, but it's been taken into the forest where these beetles have not established before. So you also have to be very careful of that. And also in terms of the monitoring, over the 5 years' time, there's an increase in the number of trees infested in those plots, about 7.5% currently. So every time you go back, another 7.5% of the trees have been impacted by this. So it's slow, but it's also actually fast, and it's accelerating over time. From these monitoring results, what you can do is quite nice. So this is a list of all the tree species that we find the beetle in. To the right of that sort of dotted line, those are species that the beetle prefer. So they attack those trees at higher numbers, not higher numbers, more of those trees are attacked than you would expect by chance if you weight everything in terms of the number of tree species or individuals, right? So the ones on the right end of that line, higher chance of getting the beetle than per chance. Anything that touches that red line is sort of at random. This is what you expect. And then things on the left of that line has the beetle in, but it's almost by accident. It's not they're not choosing those trees at all. And if you overlay that also to the trees in which the beetle breed, the number of holes that we're looking at in these trees, there's a big overlap between the previous slide and this slide. So the number of trees infested and the number of holes you find in them, which they can breed in. So it seems, at least in our systems, that they can choose trees that they breed in somehow. You can also look at the families of trees that are highly susceptible there. Fagaceae, like we know, is very important for them. Also the Fabaceae, the pea family trees, which is a really massive family of plants that's highly susceptible. So all kinds of nice things you can do. You can also see what are the reasons or what's helping them moving around in these forests. So top left. So this is the sort of the expected, the level of expectance of a tree to be infested over time. So the first graph there to the top left, the closer you are to an infested area, the more likely it is that you will get the beetle, which is great. If you didn't see that result, there would have been problems with our data. Also, as we all know, the more hosts you have, breeding hosts you have, the higher the chance is that you will get the beetle. And it's nice to actually show this and not just tell people this is what you expect. We actually have the data to show this. The higher the density of trees in the area, well, in these forests especially, the lower the chance is of getting the beetle. So these beetles like a little bit more open areas. They don't like the deep forest, deep dark forest areas to infest. So they focus their efforts sort of on the edges of the forests. And then in accordance with the biotic resistance hypothesis, the more tree species you have in an area, the less likely you are to actually deal with PSHB, the less likely they are to find a host in that area, which is nice also for urban areas. It means the more you diversify, the less chances you're going to get or have to get this beetle. This is just a number of holes in trees, so the sort of how badly individuals are attacked. So larger trees tend to be attacked at higher densities of the beetle, so there's just more space inside the tree for them to colonize. Trees with worse canopies, bad canopy health, seem to be infested at higher levels. Now this could be because of the beetles or they were in bad condition before, so we didn't actually discern between those two. Water is important, and we relate that back to the stress of the trees. So the further these trees are away from water, the higher the impact on them. So that is sort of combined for all the species, but also for individual species, we find the same thing. So what we take from that is that the happier the trees are, the healthier the trees are, the more resistant they can be for PSHB. And the last graph there is just for the increase over time. And the urban monitoring results echoes exactly what the forest results show, exactly the same patterns we see. There we also followed the numbers of the beetles over time. So this is at one sort of wine farm that we looked at. If you look at the numbers of beetles there, so that's the number of individuals in a trap over 2 weeks, and it's almost close to 14,000 individuals in a single trap. That is 100 times more than the highest in a trap in this area. So once you get to those sort of numbers, they will go for anything. Anything is seen as food. Yeah, so the poor student had to count these things. But we also see that sort of huge dip in the numbers during the colder, wetter winter months. So we have the similar climate to here. And that is very strongly temperature sort of correlated with temperatures. Right, so that's a sort of urban and native systems. South Africa is a little bit of a ground zero for a lot of the fruit trees. So we know that PSHB can have impacts in avocado, although not as severe as people thought or once thought. Pecan nuts, we have the same sort of thing in pecan nuts, that we seem to be able to manage that. Macadamia, we have seen a few dead trees, but these tend to be in places where the trees are not really managed well. It's kind of left to their own devices. Same with some of the citrus. We have not seen it in orchards and citrus. We have seen it in gardens. So I'm not sure if that's going to be a problem there. But we preemptively did a lot of sort of pathogenicity trials on stone fruit and on pome fruit, because we're in a sort of a region where that's really important. We often find this thing. So if you do science and the farmers forget that you're doing research in the orchard, you do some tests and they take out the orchard before telling you that they're doing that. So we started with some of the pathogenicity trials and they kind of took out the orchard where before we had the results. So we saw there might be some troubles heading. And then a year later, right next door to where the orchards were taken out, we saw this thing. So everywhere you see a little spot there, that's where an entry hole for a beetle. This kind of thing you can often see in a garden, but when you see this in an orchard, then you should start to worry a bit. A little bit of a closer look. If you open the bark up there, you can see the holes in there, some necrotic lesions around the holes. The fungus grows really well in there. The beetle does not do that well in there, but it's creating all of these battle scars and these wounds. And we're also seeing a lot of these secondary wood rotting fungi and things coming into these wounds in these trees. So the chances of this 7-year-old tree reaching 30 odd years old is very, very limited. We also had a quick look at some of the fruit production there. Not going to go into much detail here. Basically, the results indicate that the fruit gets riper much quicker when the plants are infested, which it's causing problems for the farmers in the sense that they have to harvest the same orchard more than once because all the fruit are not ripe at the same time. I have to also say that this is very sort of early results. They've only been in the trees for 2 or 3 years now. We have to see what's going to happen over time. Stone fruit, a little bit of a different kettle of fish. And initial trials, the pathogenicity trials said, yep, these plants can be hosts. We also see them in gardens. People plant them in the gardens. We've seen trees being killed in gardens. But in orchard settings, I mean, in South Africa, they spray everything known to man. So we didn't expect to see huge problems there. We find it in a whole bunch of hosts. Basically, anything in the genus Prunus seems to be a good host. And then we started seeing it in orchards, those little glue drops here and there. You see some infestations on them. Then things get a little bit worse as of last year. So that's I think it's a Prunus, a prune tree. And you can see from the previous wounds, the bark is already starting to split. It's a bit early for those trees to show that. And lots of new infestations on these trees, and they can breed in these hosts as well. And we're starting to see a little bit of this, which obviously, if you're a farmer, is not a good thing. There are some dead trees also there, but we can't ascribe that whether it was PSHB or not. So we're just monitoring now to see. We also have a master student looking at this now, looking at the fruit quality and looking at the export possibilities, what's happening when we actually try to export the fruit to China, which is one of our sort of main export countries. Yeah, so management, except for cultural management, physical management, everybody wants to look at chemical management as well. So we did the same in South Africa. We've published on one of the tree species, but we've done this on multiple tree species, and the result is pretty much the same. So you'll see those injections there. These are not really trained people. That's the students doing the research there. So we actually used the sort of top middle picture, these 3D printed traps to put the beetle into the tree. And we also put the fungus into the tree at a specific time so that we know exactly when these beetles infest the tree and the fungus infest the tree. So you place that in a tree, let them do their thing for about 2 or 3 weeks so they can establish their colonies if they want to establish their colonies. And then we treated these trees with a whole bunch of chemicals. You go back and you slice that branch into little discs, and you can actually follow the galleries of the beetles. You know exactly how long they had to make those. And you can count the larvae, you can count the adults, you can count the eggs, all kinds of things in there. And you can also measure the growth of the fungus in there. So some of the results, so this is looking at the beetle, once it's already established a colony in the tree. There's some of the chemicals we used, some are trunk sprays, some are sort of taken up by the plant, some are insecticides, some are just fungicides. And you can see that blue block, we're reducing the number of successful colonies in that tree, but only about by a third, which, I mean, you buy about a year, I think, in terms of impact on that tree. So to reiterate what Shannon was saying, this is not a silver bullet. It's not going to help much. You have to use this in conjunction with all the other management options. So no, you're not going to stop it like that. Can you kill the fungus when it's in the tree? Well, not with the chemicals that we've tested. You can reduce the growth quite substantially. Propiconazole is a sort of a fungicide that you inject into the tree. You're slowing it down, but if you go and isolate that fungus, it's still alive. So you're not killing it. You're just slowing it down. So that's not an option. Can you stop new attacks? So the two sort of surface sprays, insecticides that we used, again, you can slow the new colonies. This is only a month after these things have been sprayed, and they still made it through that sort of highly toxic stage of the tree. So you're going to have to keep these trees highly toxic for a very, very long time if you actually want to prevent anything from going into them, which is not an option for anyone. So that's also a resounding no for that. So we're back with the cultural sort of control options, and you'll probably have the same issues that we have. We have to prove that taking a tree down is a good idea. So we actually sort of earmarked 140 trees that we knew were going to be taken out. Some we were going to take out in winter, and some we're going to take out in summertime. And we decided to monitor the numbers of the beetles as we progress through this sort of process. So we put these traps out underneath these trees 7 days before we take the tree down. On the day of the tree, we take the tree down. We also count the beetles in that trap. And then every day for 7 days after, we also counted the beetles in these traps. So you can see there's a huge difference. Just look at the bulge of that gray area of the graph. That's sort of where most traps, or where most of the data lies for the traps. So before you cut the trees down in winter, you see there's not much happening, but there is some beetles flying around, but most of them are on zero. In summer, you can see per day, per trap, you can get more than 150. So we get in one trap, it's the same number of beetles that you, in one day, same number of beetles that you get in 2 weeks in a trap. So there's a huge difference there. On the day that you cut them down, there's an actual increase in the number of beetles that fly around. So you're disturbing those beetles. In the summer area, if you look at that bulge higher up, in winter, nothing much has changed. The beetles still stay in the tree. And then afterwards, in winter again, there's a reduction even then. But in summer, there's a massive reduction in the number of beetles flying around. And that is just for a week after that tree has been taken down. So it's almost an immediate effect. So what we can see from this is when it's nice and cold, cooler weather, that's when you take these trees down. So if you are monitoring trees in an area and you see a tree will have to be taken down, if you can wait until winter and it's cold, that's the time when you do it. Biological control. So we're also doing quite a bit. I've got a master's student and a postdoctoral student looking at biological control options. Also some PhD work that was done. So some of this work has also been conducted not in our lab. So this work has been done in Pretoria, and we're sort of taking it a little bit further. So in terms of biological control of fungi against the beetle, everything works beautifully in the lab. You can kill them quite easily. It's not a strong beetle. It's weak. But as soon as that beetle is inside a piece of wood, you can't get to it. Well, that's so we thought. More on that later. But as soon as it's in a piece of wood, and Brett would mention some of these things, this fails. And I'm saying they're not yet because there's ongoing research on that. We also often get asked why are we trying to target the fungus in terms of biological control. So we're doing that with a fungus called trichoderma. And this is it's a fungus that eats other fungi. So it loves to eat fusarium. The bottom plate there's fusarium, nice colony grain. The top plate had exactly the same colony on it, but just 7 days before we put this trichoderma on there, that green fungus, and it just killed it. It ate the fungus. Again, so in the lab, it works beautifully. Doing that on a piece of wood, the colonies of wood, it does not work at all. Nothing happens. Again, not yet. So we thought it might be a good idea. So we thought that the problem here is that these control agents not getting into the gallery system. So we thought, well, an innovative way of doing this is to try and use taxis. So in PSHB, if you look at PSHB, you catch enough of them. You also send a picture, thank you. If you catch enough of them, you'll see that there's little things crawling on them. So big fleas have small fleas. So on these beetles, you'll find these mites. Some of them might be antagonistic to the beetles. They might feed on the eggs or something. The ones that we're working with actually feed on the fusarium fungus, and others are just there to get a taxi from one tree to the next. So we got in South Africa about 7 different species of these mites, and we thought we can try and use them to take these biological control fungi into the galleries. So to do that, we need to be able to breed these things, and the ones in bold there, we actually have in culture. They eat the same fungus that the beetle eats, so we can actually grow them in high numbers. And what is nice about those particular 2 species of mites is that they also eat the biocontrol fungi. So you can grow their populations on a biological control fungi, and they actually acquire the spores, which is great. And we got to that stage, and then things started falling apart for the poor student that was doing this. So we actually have colonies of the beetles on artificial media and tubes, and we put the mites in there, and nothing seems to happen. The mites go on happily. They are full of the fungus, the biological control, but nothing happens. So more on that later. Also in conjunction with the University of Pretoria and Grahamstown, we also looked at parasitoids. So Brett will tell a little bit more detail about that a little bit later. So these are these little wasps that will fly around, find the beetles, lay the eggs on the beetles or in the beetles, and the larvae will feast Whole bunch of species. So we're looking at what is in South Africa, also a little bit of what is in the native range. So these are the wasps in the South African context, and most of these are highly likely to come from PSHB. We're still trying to figure out or trying to connect or make a definite connection between the beetle and these wasps. But the take-home message here is that a lot of our native parasitoids are actually jumping from the native beetles onto PSHB. By doing that, we get these things often. They're obviously not doing the job. The PSHB sort of breeding rate is way too fast for that. Another problem we have in sort of tackling PSHB with these parasitoids is that they're not very host specific. So in that network that you see there, all the green dots are different tree species. The orange dots or yellow dots, that's different beetle species, so native beetle species in our systems. The red dot that you see there, that's Uvulaceae fornicatus. And then the blue dots are these wasps, the parasitoids. So if you look at the red dot there, you'll see a whole bunch of blue dots connected to it, these wasps that we got from PSHB. But if you look at those blue dots, you'll also see they are connected to a whole bunch of other orange dots. So they're also associated with a whole bunch of our native beetles. So in a biological control program, this is not an ideal situation. If you add these things to the system, you're going to start impacting your natural systems. So no silver bullet there yet. This shows the same sort of thing. We've got a postdoc looking at the nematodes. So we've got 8 species of nematodes we're testing in South Africa for efficacy. So that little video clip that you see there, it's nematodes that we harvested from a PSHB beetle, killed by that nematode within 2 days. So in the lab, within 2 to 4 days, these nematodes can actually kill the beetle and their larvae, which is great. We'll just have to start taking this further to see, can we actually apply this in the field, or how to apply this? Maybe also using mites to transport them. So I said there's a little bit of a sort of a kink in the cable here. So with those experiments that we did in the lab, where we tried to put the mites with the biocontrol fungi into these artificial colonies systems, nothing happened. So we thought, well, let's just have a control experiment first to see what's happening here. So some of the tubes, we just put the fusarium in, and then some of the other tubes, you put the fusarium in and then have a female beetle create an active colony in there. And they just put the trichoderma fungus, the biological control fungus, into those tubes. When fusarium is on its own, the trichoderma wins. As soon as the beetle is there, the trichoderma does not even germinate. So we suspect, we're doing some tests on this now, that we have a similar system as with the leafcutter ants, where those ants, they harvest leaves, and they grow a fungus on that. So they also feed on the fungus. But these fungal gardens get taken over by a whole bunch of other things if the ants are not there. So they need a way to keep us clean. So they have these bacteria associated with them. These bacteria grow in the same colonies, and they actually produce antibiotics that kills other microbes in those systems, like, for instance, our trichoderma fungus. So we suspect one of the reasons why we cannot get this biocontrol into these systems is that there's this bacteria in the system that sort of facilitates, keeps the system clean. So we're actually dealing with a much bigger issue here. So we have the beetles which farm the fungus, or we could actually say the fungus farms the beetle also, because this is one organism. The fungus is the food, the tree is the host, and then we have this bacteria or possible bacterium in there that's the guy that's protecting all of this. So we should probably focus more effort on finding out what is that and can we overcome the guard in the system. So can we stop it? Definitely not in South Africa. So you really do need to stop it before it gets in. Also, new genetic material of it shouldn't get in. You don't want these things to be able to adapt to different climatic conditions. You can reduce its impact. You're trying to reduce that curve, like within COVID. You have to keep the numbers of the beetles as low as possible. As soon as you do that, you don't see the host impacts. You don't see alternative hosts being affected. You basically see your sort of main host affected, and that's the only one. Monitoring. That's the one thing that we get the most information of, and that's the one thing that people sort of skimp on in terms of money quite often. And you need that to get the information. And similar to everybody else, the problem with PSHB that we're seeing in South Africa is that pests and pathogens that came into the country before then affected one section of society. It's agriculture or forestry or natural systems, and tended to be on one host, where this beetle affects everything across these sectors. And now for the first time, these different sectors actually have to speak to each other, try to find out a sort of a common solution to a problem. And there are massive walls in South Africa in terms of that sort of speaking to each other, sharing data, sharing knowledge. There's all kinds of walls for that. And more things might be coming, so don't just focus on PSHB. In the sort of looking for PSHB, we're picking up other things as well that's not problematic at the moment, but could be, like that Amasa beetle, which is very similar to PSHB, but it infests eucalypts in South Africa now. Yeah, so I'm just going to thank a whole bunch of people and organizations for what we've been doing. Thank you.
Thanks very much for that, Francois. And I know there's another lot of questions that have come in during that talk. I'd like to introduce our third international speaker for the afternoon, and it's Professor Brett Hurley, who is joining us. And can everybody in the room please cross their fingers? He is joining us virtually from the University of Pretoria in South Africa. Professor Hurley is a forest entomologist and full professor at the Forestry and Agricultural Biotechnology Institute. His research focuses on insect pests of plantation forests with a strong emphasis on biological control. His work examines parasitoid host interactions and the ecology of nematodes to improve the effectiveness of biological control in forest systems. Today, he will discuss how he integrates ecological understanding with practical management to support sustainable forest protection, particularly in the sub-Saharan Africa, and how these lessons can be applied to long-term PSHB control strategies. Thank you.
Great, yeah, thank you again for the invite. Just to present today, I'm going to give quite a broad overview of biocontrol, the principles of biocontrol, and the challenges as well, and how it can potentially be used to manage invasive pests that attack forest trees, and then mentioning some of the work that we've been doing for the biocontrol of PSHB. Some of that's also been covered by Francois in the previous talk, but I'll just mention some of that work as well. So I work generally on the management and understanding of forest insect pests, whether invasive or native. And one of the approaches that we've often used is biocontrol, which I'll get to just now. So in South Africa, as with many other countries, including Australia, we have that challenge of a number of invasive pests, including insect pests, that have been introduced into the country over time, and also seeing that pattern of this exponential increase over the last few decades of these invasive species, not all of which become established, but some of them do become established and can cause serious economic, serious environmental, serious social issues in the country. Of course, one of those that you are very familiar with, and which is the topic of this workshop, is the Polyphagous Shot-Hole Borer. And as Francois mentioned, that's quite unique in having this massive host range and making it even more problematic. So that I'll touch on as well. But yeah, my interest is really in the research, applied research around biocontrol and general pest management, and then the implementation of solutions. So whether that's deploying biocontrol agents and post-release studies and understanding what factors influence the establishment of those biocontrol agents, or whether it's other aspects of integrated pest management and the implementation of those strategies based on the knowledge developed from applied science. But before I carry on, I just want to really give a broad background in biocontrol. There might be people in the audience that are quite familiar with this and have even been involved in biocontrol projects, and then maybe others that are not so familiar with the concept and have just heard it mentioned. The basic concept of biocontrol is that you want to introduce the natural enemy of your target pest. In this case, we're talking about insect pests, and you want to introduce that natural enemy to suppress the population of the pest to below a level where it's causing undesirable impact, whether that's economic damage or any other type of impact. You want to bring the population to a low enough level that that impact is considered not serious, not significant, etc. The key principle here is that biocontrol does not eradicate the target insect in this case. It merely suppresses a population, but sufficiently so that it's no longer considered a pest. So that's an important concept with biocontrol. It is not an eradication thing. It's not a pesticide. The pest will still be there, but in low numbers that it's not considered an issue. So that's the basic principle. And you'll still get fluctuations. The pest population will still go up now and then and down as the natural enemy population fluctuates as well. But it will be below, if successful, it will be below a level that's of concern. So just in case you are not very familiar with biocontrol, and you might have already heard or been exposed to a number of examples in previous presentations, but I'll just briefly touch on the fact that when we talk about biological control of insects, there's really a large range of potential, what we would call agents that you can use in biocontrol. So, and these are some things that we would consider, for example, with a Polyphagous Shot-Hole Borer, like would these agents be suitable, or should we be looking at these potential biocontrol agents? So, some of them that have been very successful for other insects, such as aphids, would be predators. So you get other insects that are predators. Obviously, spiders are predators, birds, etc. So there's a whole range of different predators that have been used historically to target insect pests. I mean, obviously, the one big issue here is predators are not always very specific, and that is an issue to consider if you're introducing it into the country. Parasitoids is a big group, and this has been used for a number of beetles as well. So you get parasitoids. So parasitoids are generally free-living in the adult stage. They always kill the host, and they normally have one host. Predators will consume a number of different hosts. And a lot of the parasitoids are in the order of wasps, which are called Hymenoptera, and Diptera, which are your flies. Some of these parasitoids themselves have their own parasitoid, and you obviously want to avoid that getting introduced. But parasitoids have been used very successfully in biocontrol programs, both wasps and flies. And the good thing is they're often very specific, so which makes them safer to use with regards to not then having non-target effects when you introduce your biocontrol agent, because you don't want your biocontrol agent impacting other, for example, native insects. And then you get things like entomopathogenic nematodes. So Francois touched on these. These are really fascinating organisms that have an association with bacteria. The bacteria releases toxins that kill the insect host. The infective juveniles of the nematodes then reproduce inside the host, feed on the bacteria. And there's a lot of very, very interesting work done on entomopathogenic nematodes, including in Australia, and they've been used in various systems for soil pests, other pests as well, and they can be very effective. You get things like bacteria, so you're probably familiar with BT, so Bacillus thuringiensis is used a lot, and that's also can be a very effective biopesticide. Viruses as well. Viruses often occur just naturally in the environment. They can also be used as a microbial insecticide, but in many cases we find native insect populations start to crash because of a virus that expresses itself when the population numbers increase. So this is a pine emperor moth you see in the photos, a native insect in South Africa, can feed on some of our forest species, including our pine that we use commercially, but then the population generally crashes after a while, after it builds up because of because of a virus that's naturally occurring in the system. And then you get entomopathic fungi. So there's a number of different species. There's some common genera that are used. And these, again, naturally occur in most environments. And they can be also mass-produced and used as a microbial insecticide. And this is used in different systems. Obviously, the thing here, which we'll get to later, is with things like entomopathogenic fungi, you need to be able to get to the insect. The spores need to be able to get to the insect to actually be able to be effective. So you might, if you're walking around your garden, etc., have seen insects like these in the photos, where there's been an entomopathic fungi that may have actually killed the insect that's naturally occurring in the environment. Okay, so that's just to give you an idea of some possible agents, and there's of course many more, but those are ones that are quite commonly used in biocontrol programs against insect pests. But just when we talk about biocontrol, and again, some of this might have been touched on in previous presentations, there's really different types, and it's important to understand the different types of biocontrol, the different approaches that are used. So you get, first of all, something which is called conservation biocontrol. So that would be where you already have natural enemies present in the system, but they're not really suppressing the environment sufficiently. For example, Francois mentioned the work that's been done in South Africa, where we know we expect there might be some parasitoids that have shifted onto PSHB. But even if that's the case, PSHB is still a problem in South Africa, so they're obviously not doing a sufficient job to suppress populations of PSHB. Conservation biocontrol approach would then look at that system, understand, for example, the nutritional requirements of those parasitoids that are parasitizing PSHB, understand their habitat requirements, and then try to modify the environment to suit the parasitoid. So maybe realize it needs more nutrition in the adult stage. Let's put some strips of honey or something out in the environment that it can get more nutrition, that it can survive for longer, that it will reproduce more effectively. The parasitoid population will then build up, beetle population will drop. So just by changing the environment, maybe it needs another plant host available, etc. So that would be a conservation biocontrol approach. Augmentative biocontrol is really the mass production and the mass release of a biocontrol agent. And it could be an insect, it could be an entomopathogenic nematode. But the principle is that it's a system where you can't just release it once off. You need to release it in large numbers, and you need to release it repetitively. So, for example, in a glass house, you might have a mite problem, but you can introduce predatory mites. But it won't be once off. You'll probably need to do that a number of times. For example, it could be every year or multiple times a year that you would need to do the releases. And you wouldn't release a few. You'd need to release thousands, tens of thousands, hundreds of thousands, etc. So that's an augmentative biocontrol. And obviously this system, you need to be able to mass produce your biocontrol agent. It doesn't work for all, not all agents are very easily produced in large enough numbers to use this type of approach. And it's expensive. A classical biocontrol approach is where normally the insect is non-native. For example, PSHB, you go back to the native range of where it comes from. So for PSHB, that would be in, for example, Southeast Asia. So you investigate what is actually suppressing the insect population in that region, and then you search for those natural enemies, make sure they're safe to release in your country, they're not going to have non-target effects, release it in various parts of the country, and the idea would be that it establishes and then spreads by itself and suppresses the pest population with very little effort after that initial introduction that's actually needed. That would be a classical. So obviously it needs to go into quarantine. There's a lot of host specificity tests needed, etc. So that's just generally the main biocontrol programs that are used or approaches. And then you also get something called natural biocontrol. So natural biocontrol is, when people talk about that, they're generally just talking about things that are already in the environment and might be having a very key role in suppressing the pest population. So that requires no human intervention. This is obviously first prize, when you don't actually need to do anything because something that was already there, native insect or native fungus, etc., has started to attack the invasive pest and is suppressing the population. Okay, so just to summarize, these approaches are very different. They normally target different pests. Some target more invasive pests. Some are used mainly on native pests. The investment's very different. Classical biocontrol, because you need to go back to where the insect came from and do a lot of exploration work. There's a massive cost up front generally, but it can have a very long, if successful, it's got a really long-term management of the pest with very little cost after that. Augmentatus, more of a continual cost, and conservation has a gradual time to impact. So just important to note that natural enemies or the use of biocontrol is really just one option that is used for the management of insect pests, and it's often used most successfully when it's integrated with some of the other possibilities. This is just an old slide. There's a lot of other methods now that you can use to manage insect pests, but the basic principles are that you could also use things like host resistance. You can do things like modifying the environment, pesticides, etc. And so biocontrol is not always used just by itself, although in some cases it can be, but also there's a lot of knowledge about the insect, about the general system that would help for biocontrol or any pest management approach to be successful. You need to understand when is the insect occurring, understand its biology, when would be the best time to release the biocontrol agent, the genetic variation of the insects, etc. So it's part of a broader integrated pest management approach where you look at potentially using biocontrol. And biocontrol is not always the best approach to use for a specific insect, and it's not always going to be successful even if you do find a biocontrol agent and release it. And this is partly because not all insects are, the population of insects is not always controlled mainly by what we call top-down forces, so like natural enemies that suppress the population by parasitizing them or preying on them. In some cases, it's actually more resource availability that's the main driver of insect population. If the resources are available, if there's susceptible trees that are planted in the environment, the population will maintain or it will grow, the insect population. But in other cases, top-down forces like the introduction or presence of natural enemies is actually a major regulating factor of the insect population. And in many cases, you'll find that both bottom-up and top-down forces are important. The degree to which they regulate the population is very important to understand, but quite difficult sometimes to actually understand. But this is important work that we should be doing with our invasive insect pest species. Understanding the population ecology or the population dynamics of those and how natural enemies and how bottom-up forces like host resistance or host susceptibility, what role they actually play in the population or the size of the population. When it comes to the biocontrol of the Polyphagous Shot-Hole Borer, is this going to be a really viable solution? That's the big question. And one of the positive things towards that question is that actually biocontrol has worked quite well with insect pests of trees. I mean, it's not all cases. You can see here maybe 1 out of 3 that it's become established or where the target's been controlled, but that's better than in many other systems. And I think this is partly because forest trees just provide a more stable system for the parasite or for the natural enemies to actually become established, as opposed to, for example, an agricultural system that's constantly harvested, or at least more frequently. There's more turnover in the system. And there's been a number of success stories of biocontrol of forest insects, many actually. One of the ones that I want to touch on is about the Syrinx wood wasp. The Syrinx wood wasp, the reason I'm giving this as an example is it's quite similar in some aspects to the Polyphagous Shot-Hole Borer, and it's also a wood-boring insect. So the larvae of this wasp lives inside the tree, and it also has a very important symbiotic association with the fungus. In this case, really, it's just with one species of fungus, so a bit different, but a lot of similarities as well. And there's been a biocontrol program that was developed, and actually it was developed, well, mostly in Australia, really, and optimized in other countries over a number of years. This is Robyn Bedding, who was kind of the pioneer behind using this parasitic nematode that was then used to sterilize the wasps, and really been very successful to suppress the populations of this wasp, not just in Australia, but really around the world, and especially in the Southern Hemisphere, it's been a major success story. And this is just quickly showing you some photos. So this is the Syrinx wasp, kills pine trees, introduces symbiotic fungus and some toxins, and here's the larvae boring in there, and the nematode would sterilize that and help to suppress the population of this wasp. And here we have some just a diagram showing the number of parasitoid wasps were also introduced. In this case, it was multiple biocontrol agents released, and the whole biocontrol program was then established. We had to, well, in Australia, they developed basically how to rear the nematode using, because the nematode feeds on the same fungus that the wasp introduces. So a whole system was developed with regards to mass producing, mass releases of the nematode, preservation of the strains of the nematode in liquid nitrogen, etc. And this has been adopted by different countries, optimized, and been very successful. So this is just in South Africa. So we do this every year. We're doing inoculations of these parasitic nematodes to suppress Syrinx populations, and we, producing billions every year that go into the field. And it's been highly successful, saving South African pine industry hundreds of millions of rands. So really a highly successful biocontrol program that I said has some similarities to PSHB. It is important to note that, even though this has been a successful biocontrol program, so our populations of Syrinx are much lower in South Africa now, it's not necessarily just because of these natural enemies that have been released. There's also other factors that have changed the environment, and a number of different things can actually influence the population. We need to understand that better. That includes, the availability of susceptible hosts in the environment. That can also help to regulate the growth, competition of the organisms, etc. So the main point I'm trying to make here is that it's really important to understand what factors influence population growth of a pest species, to know, or to be able to predict when you might get outbreaks, even in the presence of a biocontrol agent. So when you're going about considering implementing or developing a biocontrol program, there's a number of considerations, and there's a lot written about this. This is just one example of a guide. So Simon Lawson and myself and a lot of others were involved in this. This is an FAO guide. Mark Hennis led this work from KEBI. Some of the considerations would be things like regulations. So you need to understand the regulatory framework in the country, in this case Australia. Are you even going to be allowed to introduce something like a classical biocontrol agent? What are the requirements going to be? And to make sure that you're considering those when you're deciding what biocontrol option you're going to use. You want to see if there are existing biocontrol programs, because that would obviously be first prize. That, like, for example, for the Syrinx wood wasp, when we had Syrinx introduced in South Africa, we didn't need to reinvent the wheel. We could adopt what was already developed in Australia and then just optimize it for our own country and make some changes, and that's been successful. That's actually been a really successful case of different countries adopting an existing biocontrol program. You want to see if there's already biocontrol agents in your country, so native insects, etc., that have actually shifted or expanded their host to the target insect, so for example Shot-Hole Borer, and maybe already starting to do a job of suppressing that population. That's really important to know. And then you also can consider looking at classical biocontrol, where you'd go into the native range of the pest and look for natural enemies to introduce. And when you do go down that route of looking at a classical biocontrol approach, there's a lot of things to consider. Where you're collecting the insects from, is that climate similar to where you want to introduce the insects, because that can then have an effect, the biocontrol agent. The genetics of the biocontrol agent, the genetics of the pest. Are you sure there's not cryptic species involved? So that's really important as well. And obviously host prey specificity. So for biocontrol, really a big thing is you must be sure that it's specific to your target pest. And those studies are not necessarily that easy to do. First of all, you need to have a good understanding of what the potential non-target insects could be. So you need to have a good understanding of the fauna and the biology of that. You need to be able to rear those to be able to do the host specificity tests. And yeah, that could take years. Sometimes this is the major barrier with classical biocontrol, is getting through all the host specificity tests that need to be done. and to come up with results that you can be confident in, and that people that you send this to review are confident in your results. But just mentioning some of the work that we've done with biocontrol at PSHB, and I'm not going to be too long on this because I know Francois touched on some of this work, because a lot of the initial work we worked together on this. But yeah, PSHB, I think you've been giving enough background on that. But just to say that the work that we've been involved with was funded by what was then the Department of Forest, Fisheries and Environment. The Centre for Biocontrol was a key partner, as was Stellenbosch University. And we did a lot of the exploration work in Vietnam, and we had very good collaborators there at the Vietnamese Academy of Forestry Science, and then obviously FABI, the institute where I'm from. So this is just to show you some photos. So you would have seen Garen's photo in some of Francois' slides. So he was a PhD student at this time and was helping with some of the exploration work. And there's Wilma Null, second from the left, and there's our Vietnamese collaborators as well. And we went both to northern Vietnam and to central Vietnam on multiple trips, doing collections in the field, looking for where we could find PSHB in that environment, and then bringing that material back to South Africa into our quarantine facilities. So we brought back, I think, over 200 kilograms of infested wood. This was over 2 trips, and then we looked to see what would come out of that material, with the hope that we would get a parasitoid or some type of potential biocontrol agent that we could use. So this is, this type of approach is a bit like looking for like a needle in a haystack. The more effort you spend, the more likely you are to find a potential candidate. So you don't know exactly where you should look, what host, you don't know the biology of the parasitoids, etc. And this is just us looking at some different plant hosts here on acacia, and here on castor bean. And this is us in our trendy little quarantine suits back in South Africa, where we've brought all the material back, and we're now looking to see what emerges from our emergence boxes. And then we had a number of different insects, a number of different wasps specifically we're really looking for a parasitoid wasp. And we had a number that actually emerged, many of them with, no names and not necessarily easy to describe, and some of them really tiny. So this mammarid wasp is about 0.2 millimeters. It's extremely small, very difficult to work with when you want to now do tests to see, is it definitely a parasitoid of PSHB, because it could be other insects in those logs we brought back. So it was very difficult to work with these insects. But just to cut a long story short, unfortunately, although we went through all the rearing process of these parasitoids, exposing them to PSHB, etc., in the end, we came to the conclusion that they were not, we couldn't confirm that any of these parasitoid wasps were actually biocontrol agents of PSHB. So we had some potential ones, but in the end, after the test exposing them to colonies of PSHB, we found no parasitism of those beetles. So most likely they were actually parasitizing another insect that was present in the material that we collected. We did, however, find a nematode in the beetle populations from Vietnam, which was very interesting. It's something that we're considering further work on. The only thing is this nematode maintains itself in the colony of PSHB, and it also feeds on the symbiotic fungus, but we haven't observed any mortality of the PSHB. So that's not really giving us great confidence that this would be a good biocontrol agent. But that is a potential avenue to understand the biology of this nematode a bit better, and if it's having any fitness, reduced fitness on PSHB colonies. We've also engaged with USDA previously, and were ahead of us. They've actually done a lot of work in Taiwan looking at potential biocontrol agents, and they did find a parasitic wasp that offered a lot of promise. So there's definitely some potential with regards to this classical biocontrol approach. Then Garen, as part of his PhD, was looking at natural enemies in South Africa that might have shifted host to PSHB. He collected a whole lot of material, and Francois was presented on this already a little bit. But the basic thing here is, is there something in South Africa that's already shifted and parasitizing or preying on PSHB? So this would be obviously an important thing in Australia, and you've probably been doing this through your monitoring already to understand, are there already things out there? It might be an intimate pathogen, and could those be used then? Because then you know that PSHB is a host. Then obviously, the fact is it's not at that moment suppressing the population, but could you augment that? Maybe it's a parasitic wasp or an intimate pathogen that you're able to augment and do now mass production, mass releases, or maybe it's something you could use a conservation biocontrol approach for, without having to then introduce a foreign insect or a foreign biocontrol agent into your environment. So this is definitely something where it's worthwhile to keep monitoring to see if there are things that are naturally moving onto PSHB. So this is just Garen's work. Again, Francois presented on this. He really found a lot of things, a lot of different wasp occurring. We also got a better understanding of our native ambrosia beetles and their parasitoids, because he wasn't just targeting PSHB. So that's really good to that understanding, because if we were to introduce a biocontrol agent, this would help us to understand what some of the non-target effects could be that we would need to test for. And he found some that could potentially be parasitoids of PSHB, that also needs a bit more further investigation to see if they would be suitable biocontrol agents. So that's just a summary of that work, and that involved Francois Martin-Hill from Rhodes University, and was really led by Garen Townsend, who is now a postdoc with Francois. Okay, and then again, we did some work. Francois actually gone into a lot more detail about this, but just to say that intimate pathogens could potentially also be used. So, for example, fungi, you could use bacteria as well, and they work very well in the lab. I think Francois mentioned this in his presentation. Generally, these type of things, if you directly apply intimate pathogen to an insect, generally it works very effectively. You can get 90–100% mortality. And that's pretty much what we got here. We got really quick response from using these intimate pathogens, as these are just survival probabilities where you see how quickly the beetle population declines when you apply these intimate pathogens, especially the 2 fungal species that we used. These were just commercially available products, but you could actually use things that you've directly isolated with PSHB in the field as well. But the challenge is when you apply it onto the logs, actually getting it to the target insect is difficult. And this is where I think Francois is now trying to carry on to see, is there some type of novel application method that you could use to use something like an intimate pathogen, like as a viable biocontrol approach. Obviously, you'd also need to consider, the cost implications, etc. So in a natural forest, this is probably not going to be a viable method anyway, but perhaps in an orchard or in an urban environment, the cost might be well motivated to use such a method. And this is just showing that, unfortunately, with our initial test, but we didn't go too far down this avenue, we were not able to successfully use intimate pathogens when applied to infested logs. And then the main point I want to make from this slide is what we're really going into is a bit of a different approach now, where we're trying to see what are the volatiles that are released by the fungal species associated with this beetle, with PSHB. So there's volatiles that the fungi, there's some symbiotic fungi, but also the fungi that kill PSHB that are released. We want to characterize these and then look at whether they have a behavioral, whether they induce a behavioral response from PSHB. So, for example, you would expect or hypothesize that an intimate pathogenic fungi, the volatiles from that might cause a deterrent behavior or a repellent behavior in PSHB in the beetle, or a symbiotic fungi, the volatiles from that, there may be an attractant behavior from the beetle. And then if you can synthesize those volatiles, you could potentially use it for some form of behavioral control. So that's just an avenue that we're starting to explore, and we're collaborating with people on this project. We've done some initial work, but there's still a lot to do with that approach. So just to conclude, and some thoughts around biocontrol of PSHB. These are just some personal thoughts. So I think like a classical biocontrol, so that's when we're going into where PSHB originally comes from in Southeast Asia, that remains a viable option. When you go and you're doing exploration work, looking to see what natural enemies are there. But you do need to consider various factors. You need to understand the regulatory framework, climate matching, making sure what you're going to be introducing is actually going to be effective, etc. And there's a lot of work. I mean, this classical biocontrol can take many years and quite a big investment. It's expensive to go to different countries. Also requires really good networks and collaboration in those countries. You need to be working with the right people. You need to make sure you can bring the material out, etc. But there's been many cases of successful classical biocontrol programs. So that gives us some hope. Other biocontrol options using entomopathogens, nematodes. Francois mentioned mites. You know, all of those offer potential. There's a massive challenge with an insect like PSHB around how you actually would apply these type of options. So classical biocontrol, you know, that would, like a parasitic wasp, it would be introduced in the environment. It's already a natural enemy of that pest. So the biology, the host searching, getting to its host, that's already sorted out through evolution over many years. When we're trying to use something else that's maybe not a natural enemy of that pest, for example, an entopathogenic nematode that hasn't previously been associated with PSHB, you know, the big thing is that actual mechanism of it, like host location, etc., is not there. So we need to find a way of applying it so it can meet the target insect, and that's a big challenge with something like PSHB. But that's just something. Doesn't mean it's impossible. It just means that it's not an out-of-the-box solution. You really got to need to think of something novel, and that I can see Francois was doing some work around that, and it is something that's worth exploring, I think. Definitely continued monitoring is needed to see if there are going to be things in the environment in Australia, in this case, that actually start to infect PSHB and could be used in a biocontrol program, either augmentative or conservation. So that, in my opinion, is definitely worth monitoring and assessing. And then I think just in summary, my point of view, and I'm not an expert on PSHB, my knowledge is more just broadly working in forest insect pests, but when I look at this particular problem, particular invasive species, it's definitely not a low-hanging fruit with regards to using biocontrol. This is a difficult insect to manage, not just using biocontrol, but all other approaches as well. And it's going to be, I think it's going to be a bit challenging, but I think there are definitely possibilities, and most likely is going to require an integrated strategy, and also using some novel approaches, possibly developing some new approaches that haven't been used before within a biocontrol context. So I think there's possibilities. I think it's going to take probably a lot of collaboration as well. And there's a number of countries that are working on this, so that also gives us some hope. And there is some initial work that's been done. And as I've given that case study of Cyrex, where you can see that was also a very difficult insect and system to manage, but through some really hard and innovative work, a very successful biocontrol system has been developed that's now used successfully across the world. And that same thing could happen with PSHB. And just with that, thank you very much. And yeah, I'm open for any questions.
Thank you very much, Professor Hurley. Brett, we've had a couple of questions that have come in through Mentimeter, and I'll ask those on behalf of the group here. Would you expect a forest biocontrol agent to be as effective where you have a multi-host urban or peri-urban environment to manage?
Yeah, I think that's a great question. So I think in the case of PSHB, that's definitely something that's going to need to be considered, is the role, for example, of host cues, of the plant host cues, with the natural enemy. So if you are introducing a parasitic wasp to control PSHB, and that parasitic wasp was found to be a natural enemy of PSHB, but on a certain plant host, and it actually uses that plant host to get to the vicinity of the beetle, then that would be a barrier for that natural enemy to be effective when PSHB is attacking other plant hosts. So you'd need to consider things like that. If plant host is not a big cue for that natural enemy to use, then it wouldn't be a big consideration. So that's again about understanding the system that you're working with and the natural enemy that you want to introduce.
And the second question, I'm hoping isn't leading down to anything political. According to the Nagoya protocols, we may have some difficulties with importing biocontrol agents and accessing and benefit sharing. Do you think there's likely to be any problems or issues that arise if we do develop a biocontrol agent and then being able to share them among different countries?
Yeah, so I mean, that's something that I'm not an expert on the situation and the regulatory framework that applies to Australia, but it's definitely something that needs to be considered before going down that route. You'd want to understand those type of potential barriers or challenges or just requirements. It's often just about meeting the requirements with regards to introducing biocontrol. And generally those things are, they're very important. You don't want to just be introducing something that might have a non-target effect. But as I said, I'm not an expert on the framework that's present in Australia, but there's obviously a massive biocontrol community in Australia that would be able to advise on that.
Thanks very much. And very tempted, well, difficult to ask a question like that as an Australian when some of our best weeds here are known as Cape Lilac and Cape Tulip and, another a number of biocontrol agents that haven't necessarily worked before. They were the questions that had come through. So Professor Hurley, we'd like to thank you very much for joining us. I recommend now that you either go and catch up on some sleep or have breakfast, because we do realise that this was a horrible time difference for you to deal with. And thank you very much for joining us again today.
My pleasure.
Next, we have Rohan Prince, who's DPIRD's Director of Horticulture. Rohan brings a vital economic perspective to the room this afternoon, representing an industry that contributes over $3.5 billion to the WA economy and supports over 14,500 regional jobs. He'll be discussing how growers can take control at the farm gate and why biosecurity is a continuum from the border to the orchard. Rohan.
Thanks very much, everyone. I'm going to try and whip through my presentation quite quickly because the experts in the room that actually know what they're talking about in terms of the biology, ecology, entomology, pathology have done a great job. So thanks very much, Shannon, for setting up. I wish I had some of your pictures. So yeah, as you heard, why I'm so invested in is because what we seek is a robust and resilient horticulture industry in WA and Australia. And we know that it's more than just the industry itself, it's the food that it produces, and we need to be able to support that vibrant community that those industries support. Helps our diversified economy, but really it's about that food production. We need our guys to be sustainable, and that means being economically sustainable. So when a pest comes in, it really puts a challenge to horticultural industries. The other thing about Western Australia, we produce food from north to south. We're kind of narrow. We've got mangoes right down south is 90% of the production of the horticulture value in the state. It's a huge regional spread, massive climate variability. And we've got this new pathogen and new pest that we really don't know how it's going to act. We can look at the other places around the world and see what we might think, but in reality, we don't know yet, guys. So the context of this response, very much that environmental, social pests, by the way, and it affects production horticulture for food. And that's how it's kind of been responded to at the moment. And rightfully so. It's hit us in the metropolitan area, affects a whole lot of LGAs. But what we can't forget is that, this is a pest that's going to be infecting horticulture from a food production point of view. There are far more trees in the natural environment in Perth than backyards as well. I mean, obviously, that's why we had to respond the way we responded. But now it's time to get ready, get prepared, and start getting ahead of it for the horticultural industry. And the funding for the response is absolutely that state commonwealth. We haven't had that industry funding yet, which creates, I think, an opportunity to really focus on what the horticultural industries that pay those levies that really want to invest in solutions could do. So we've seen this. I'm not going to go through too much. We know that we've got horticultural pest species, horticultural host species, avocado, citrus, figs, mangoes, mulberries, pears, stone fruit, and others. We know that susceptibility goes from being, highly susceptible and reproductive through to non-reproductive and low susceptibility and negligible impacts. This is all based on either observations through our experience where we've heard there's multi-species out in the Perth metropolitan area, we're not looking at it from a monoculture yet, or it's been observed in a monoculture situation in another part of the world. So we really have to take this for what it is. It's the best knowledge we have in our environment and our situation to date. Doesn't mean it's going to hold true when we get into our environments, into our production areas. What's really important here is, guys, one tree crop and its impact or its management of that tree crop can affect another. We've heard we don't choose our neighbours, right? But you know, those trees next to each other can really impact each other and the way they're managed. So on-farm biosecurity, what does it mean? Well, we all know that biosecurity in itself is that continuum. It starts at that border, goes through to the farm gate, and at every stage, you know, it's actions that actually result in biosecurity. It's not having a sign on your gate. It's actually doing the practices, making the changes and taking the steps to reduce your risk and protect your business and your industry. So that's a really important part, is take control of your own farm. You know, assess those risks that are occurring. Like, one thing we could start doing in horticulture is map, with what we know now and with all the host tree status, we could actually map not only the production crops we have on the properties, but all those non-production trees. What are they going to create in terms of a risk or an opportunity to monitor the introduction or the occurrence of the PSHB? So, you know, your box elder maples, your London planes, your robinias, all those trees that are not so far away in community gardens or gardens in the towns where you grow down south, there could be a great asset in early warning systems or awareness that you now have to start increasing your level of intervention and intervention of management to ensure that you can manage this pest. So what can we do? We can check, monitor and stay informed. Most importantly, the transition to management, I believe, will provide that opportunity to stay well informed of any changes in information that occur. So my advice is, don't be a passenger, get actively involved and invested in getting updates. Subscribe to the newsletter, subscribe to the people that can help you provide or get that information. So another thing is about you can control what you can control on your own farm, right? But then there's the influence, the risk beyond the farm gate approach. So what I've learned from industry over many, many years of working with the horticultural production industry is they know a hell of a lot more than me. They're really clever people, and what they do is they will highlight things that we don't know. So by going out and listening to the growers, listening to the farmers, listening to the associations, what they'll tell you is some of the processes and practices that just we did not think of or didn't consider as one of the risks. A good example today, as a conversation I was having, was, well, what happens when we replace orchards now? What do we do with all the trees that we're going to pull out? What risk does that create? Common practice is to do some burning and that. We're going to spread PSHB, X, Y, Z. So there's a hell of a lot of practices that we don't know or haven't considered in the context of PSHB when it arrives in a region that we really need to start thinking about. So what I'll be doing with my team is starting to work with the industries on learning all the practices that might contribute to a risk of PSHB when it becomes the area. And what we can start doing is start putting practices in place and practice them until that pest arrives. It becomes common practice then. So that when it arrives, it's not such a different and huge radical change. That's the stuff we can control. Working together as an industry, making those farmers' practices aware to the people in the response, so they consider what best body of knowledge, contact overseas specialists to see how they've dealt with it. And work with industries that might not be hosts, but could increase your risk. Like, so the classic one is that mulch. I mean, if you don't know where your farm mulch is actually being mulched from, not where it's being sold from, but where it was being mulched and moved to, I mean, I wouldn't be bringing it on farm. But you really have to get actively involved in understanding your risk and managing it. So, things we know it can accelerate. Our major horticultural production area is only about 300k south. I just heard today that means that it's not going to be 30 years before it gets down south. It could be 3 hours, moving at 100 k's an hour, not 70 miles. Sorry, but 100 k's an hour, we can get there in 3 hours, and we know that that's a highway and a pathway. So we need to start making sure that we represent from a horticultural production and food point of view, the importance of raising the awareness of the risk that other people are creating to our food industries. That I reckon everyone cares about the food industries because they love going into the shops and they love being able to get the food at a price that's affordable at the time. This will change the economic status of our horticultural industry if we're not careful and if we're not managed well. So steps to remember. I mean, starting to do that monthly monitoring of production and landscape trees around your area, working in with the surveillance grid that's occurring in the department and being aware of what's happening in that, but also doing it as a industry. This is about efficiency here and effectiveness. How can we do this efficiently and effectively? Well, it's probably through collaboration and awareness, not massive lots of individual people doing like a regional scale monitoring program. On your farm, absolutely take control, but this is an opportunity I see for the industries to really get together on this. Clean it, you know, come clean, go clean is an expression that was there. Anything you bring onto your farm, bring it in clean, make sure that when it leaves, it is clean as well. We don't want to spread the risk, being unaware of that risk. So we really need to start taking control. Not just having the farm biosecurity sign on your farm, but actually doing the practices as well. The last thing that we also heard is this bug is so similar to so many other borers and bugs. So, you know, reporting this suspect borer through the MyPest Guide app may be helpful. I mean, I don't know about you, but I find it pretty hard to guesstimate 0.85 mil, was it? That's an 0.85 mil is an accurate thing. So I'll get my pen and I'll chuck it in, says that's about 0.85 mil. I think I've got a polyphagous. What you can do is, what you definitely should be doing, I should say, is reporting to that MyPest Guide reporter app. And that is actually in line with the Biosecurity and Agricultural Management Act of reporting suspect pests. So I guess my main message here, and I really do thank the previous speakers for the such rich information that they've provided, and I'm just trying to pull the threads together here, and basically say that, you know, now we have an opportunity from where we are in Perth and the outer metro suburbs where the polyphagous has been detected to our really important production regions to learn so much along the way, and make that gap between the impact in Perth and the impact in those production regions as long as possible. And we can only do that through good practices, participation, active participation in biosecurity, active participation in assessing the risks and coming up with good practice, best practice management. So that's pretty much it for me, but thank you very much. And like I said, I think there's, regardless of the horticultural host status at the moment, we really need to keep this front of mind. I've got a couple of people in my team that are sort of leading the contact with industry, and obviously everyone's got my contact details in WA from a hort industry point of view. But Rochelle Johnson and Sang are our main contacts. So if you are in the food production horticultural industries, if you've got some really important knowledge or some concerns, do reach out. We can work in with the response team, and we can do that two-way conduit to make sure that we're getting the right information in the right context back to the response and trying to provide them information to populate that good practice guide that will be, you know, one of the products out of that response. So I think that's it from me. So everyone wants that robust and resilient WA horticulture industry, or that Australian horticultural industry as well. So thank you very much for your time today, and I hope I got us back on track with time. Thanks.
Our next speaker is Chelsea Payne, and Chelsea's Curator of Arboriculture at the Botanic Gardens and Kings Park Authority, and she'll provide us with a case study of the PSHB outbreak in Kings Park.
I haven't even met you. All right. Thank you, everyone. Last presenter of the day. I know everyone's probably waning a little bit and we have morning tea after this. Also, thank you to everyone who is online and has hung in, especially folks on the East Coast where it's getting later in the day. My name's Chelsea. I'm the Curator of Arboriculture for BGPA. Botanic Gardens and Parks Authority, and we're one of the statutory authorities under the Department of Biodiversity, Conservation and Attractions. And I think Kings Park, where this photo is taken, probably encompasses a lot of the biodiversity, conservation and attractions that we have in WA. We're on Whadjuk land at Kings Park, just up the hill here. But one of the privileges that we do have as the State Botanic Garden is that we get to care for plants from country all over Western Australia, which is an incredible privilege, but also a huge responsibility. So who are we as an agency? Why am I standing up here? Well, Kings Park is that green patch on kind of the centre of the bubble in the top right-hand corner. We're the State Botanic Garden. We're about 400 hectares, and we have another 400 hectares out closer to the coast of Class A reserve that encompasses some of the most significant cultural sites, environmental sites, and an incredible collection of plants. We were also one of the early sites that was infested with PSHB and where it was detected. I'm not going to go into that too heavily because most people in the room have probably got sick of me talking about our site and our problems. There is also a lot of other media out there for people who might not be familiar with how PSHB has impacted Kings Park. This was the first tree that was removed at our site, however. And in Perth, I've got to say, we actually haven't seen many trees die from PSHB. This tree pretty much died. And you can see on the left here in 2001, early in 2001, that was looking pretty good on Google Street View. We had some crown dieback in 2022. It went into dormancy over at the end of winter, and it simply didn't come back out. Now, this was a really significant tree. It's right at the guts of our botanic garden, and it was one of the remnants of our original South African collection. It was removed. That's one tree. We also, for most of you, will be familiar with a very large site where the canopy within this red zone was removed due to PSHB infestation, and also the complexities of a really, really difficult site to manage. Again, I'm not going to go into that too much because you're probably all bored of hearing about that. But essentially, we had a site that was left like this. And if you want to play Spot the Sad Arborist, I'm on the right-hand side of that stump there. So we've had a really significant impact in WA's most visited tourism destination. We have over 5 million visitors a year, and we're known for protecting plants. I certainly didn't become the curator there to be cutting trees down. And so I'm not going to go too much into the detail. We've had some fantastic presenters today who have gone into that technical detail around PSHB, but especially looking forward to our colleagues in the East Coast, or for people who are landowners in WA who maybe not have encountered PSHB yet. I want to talk about our experiences and the lessons I've learned, and especially how they've been centred around our function as a botanic garden. So botanic gardens are these really special places. We're plant experts, so we're plant nerds. We love thinking about plants. We love working with scientists about plants. We love worrying about plants. And generally, we're not in botanic gardens for the cash. So that means that we have to be really tricky about begging, borrowing and stealing resources, networking, and then achieving some really special things. The other thing that we do, and is a bit of a superpower, is that we connect people and plants. And as a government agency, we actually have a storefront that people like going to. And for a lot of government agencies, they don't have that. What that does mean, however, is that we have a high level of scrutiny. We have a high level of really diverse stakeholders, and we have differing expectations on us as an agency. So in 2021, we had the first detection of PSHB on our lands. We knew it was going to be bad because as a botanic garden, we had been participating in one of the Plant Health Australia Sentinel programs for some time, and PSHB had been on that list. And there were a few choice words. There was a lot of phone calls made when we first heard that we had PSHB, but we actually took lessons from other responses that we've been part of. And PSHB is only one of the biosecurity threats to our ecological communities, to our plant production, to conservation, and our urban forests. And one of the things that we had actually learnt a lot from, and hopefully I'm sure there's a lot of colleagues on the East Coast that might be listening to this who have been very generous in their time and knowledge in upskilling us in Western Australia for the effects of myrtle rust. And so again, the things that we get to do as Botanic Gardens are really cool. We get to worry about the species that we know will be heavily impacted by myrtle rust. It was first detected in New South Wales in 2010. And to date now, there's about 15 native species which are under threat of extinction from myrtle rust. It was something that was detected in the commercial industry and very rapidly moved into natural systems. So we know that biosecurity is a threat. We know from the myrtle rust response that generally we say there's about 10 years to get from coast to coast for one of these pathogens. We're about halfway there for PSHB in that timeline. So we know from that we need to upskill our practitioners at every single level, our operational staff early. We need to network and understand the science. And then through that, we can use our modelling and our scientific understanding to undertake conservation action on known susceptible species. We have a seed bank. And so one of the things that we do is we've been able to model the species in Western Australia that we know will be impacted by myrtle rust and then bring them into collection, which is cool. We got to go on a helicopter. So we'd learn a lot from the myrtle rust response. And a lot of the colleagues that we connect with regularly have taught us a lot about getting through one of those responses. And number one is network. And a lot of the people in the room today and online are a part of an incredible organic network who are able to respond to things as they come up. One of the strongest things we can do, especially in areas where there is uncertainty and scarcity in resources, is that we can build communities of practice where we can share resources and knowledge. One of the things that we can also do in that space, and it's difficult when we have stakeholders that are the public, is that by doing that and having those networks, we can create space where we can have longer conversations. We can acknowledge the uncertainty and we can provide some of the context that comes with some of these responses that maybe you can't put on a single A4 front and back piece of paper. The other thing to acknowledge in these networks is that peer support is really critical, and some of these responses really suck. So that photo of me next to the stump, the reason that I ran up that hill and was standing there with my hands on my hips was because I was actually having a little cry, because it really, really does suck. And when we're talking about people's lives, livelihoods, if you're in agriculture, when we're talking about culturally significant locations, or when we're talking about really, really important ecological systems, the distress that we're seeing in the public is real and we need to acknowledge it, but we also need to support our peers and people who are dealing with it every day. One of the incredible things that I have to thank some of our leaders for within our organisation was having an organisational vision. So having a clear policy and strategy early helps to be able to set your course. For us as an agency, our purpose is to protect and enhance biodiversity. So that means that we had our compass needle pointing in a particular direction. It means that you can engage early and you can actually advocate for your patch. We're in Botanic Gardens. What we need to advocate for is going to look different to what people in agriculture, in urban forestry and LGAs might need to advocate for, but we can all push in the same direction. I would really recommend having your own communications plan, because again, it means that you can communicate within your own organisation and externally about what you're doing and why you're doing it. And the other thing is really please use expert advice to advocate for best practice tree management. We're not very good in Australia at actually managing long-lived plants. And a lot of the responses that we've gone through biosecurity in the past haven't been around urban trees. There is a network of experts out there called arborists who can actually inform the way that you're managing your trees to mean that they have the best chance at recovering through a PSHB infestation. One of the things that we did and we were able to do was to maintain an outward focus. So you need to look at your local actions that you can do, your stakeholders that you can engage with. Tell the story. I really don't actually like being on camera, but I kind of got dragged into it a lot. And part of that is because, again, as a botanic garden, we're a bit of a trusted voice. However, we were able to tell that story about what was happening and what was going to happen next. And the next thing is don't waste a crisis. So this is not the only threatening process that is facing our ecological systems, our plants and our biodiversity in Australia. We need to tell that story. It's really important because it supports all of us. Work on integrated pest management principles. We've had a lot of discussion around that today, and that is everything from monitoring, treatment, communications, improving the health of your collection. And if you don't have access to an integrated pest management strategy and plan, make one. Lean on those networks that you have and the experts that you are in your orbit to make one that's specific for you. It's really important to have an IPM strategy and plan that you can communicate upwards to people who are allocating funding and are allowing you to go out there and make those media statements. But also it's really important to be able to communicate downwards to your practitioners. If it's an LGA, it's really important that the people who are driving around your parks picking up fallen branches know what to look for and know what to do when they find something that's unusual. And without those IPM plans, kind of saying, oh, well, we'll just see what happens. We'll wait for one to come to us. You sometimes need to pick that up and do it yourself. One of the other superpowers of a botanic garden is that we manage our plants as a collection. And what that means is that we actually have to prioritise our plants due to significance, to value, and treat them accordingly. By doing that, you can either use things intentionally as a sacrificial sentinel species. You can move things out of collection. So one of the things that we have done, which has allowed us to reduce the pest load on our site, is that we have removed trees strategically, which are not infested, but are known highly susceptible species and also tend to be weed species. So in our bushland areas where we have things like robinias or figs that have self-sown, I can't tell you how excited our bushland manager was when I told him that we were actually going to fund removing those species. Now, not everyone can do that, and it's not applicable to every collection out there. However, use that collection management. It also means that you can develop succession plans. You can focus on diversifying your collection, your urban forest, your community park. But it also it gives you the data to be able to inform those decisions. The other thing about thinking of your trees as collections is that you can start thinking of them as assets. And our trees are one of the very few asset classes in Australia that actually appreciate. So assets in a collection that you can then manage. It's really important to manage ecological communities and not just individual trees. So trees don't exist in a vacuum, and they are fundamental to a lot of the communities that we live in, not just ecological communities, but the communities that we are part of as well. I love putting this photo in. This was at the site where that large fig came out. We've done a lot of restoration work, having a really strong restoration focus there. And this is the little fauna trap we have. And very quickly, we had native species coming back to that site. So we need to consider not just that individual tree that's infested with PSHB, but how we can make those entire systems more resilient, but also the impact that potentially our treatments of those trees might have on those systems. In Western Australia, we're seeing a real proliferation of PSHB through our wetland systems, and there are areas where those non-targeted pesticides can absolutely have a devastating impact on some of those ecological communities. So if we essentially lose sight of the forest for the individual tree, we can really get into strife. I'm just going to... All right. So one of the things that we really need to do is prepare for the future. As a botanic garden, as I said, we are really worried about plants, and we're really worried about their future. I've got this image here. It's one of our incredible Melaleuca raphiophila ecosystems. For people in the room, you're probably familiar with the keystone function that they have within the southwest. For folks that maybe are over on the east coast, these are the keystone trees of our southwest wetland systems. They are systems that are already under a huge amount of pressure from other threats. We have all of those things that we know are facing our ecological communities, and PSHB is really emerging as one of the significant threats for these ecosystems. And like myrtle rust, we can go from species being really widespread to being at risk of extinction in a very short amount of time. So we really need to lean on those networks. We need to educate our stakeholders. We need to beg, borrow and steal, and continue, please, working together to do the research, to engage with stakeholders, to warn people about biosecurity, and also for those folks on the East Coast to learn from us. We get to pay it forward. What I know my organisation has learned from everyone who's been working on those myrtle rust responses, to then be able to reach out to all of us on the West Coast and prepare for when PSHB may end up on the East Coast. There's a few resources there. My email address is there. Please keep bugging me if you've got a question. If people do have an interest in the Mount Eliza Escarpment Project, we're putting all of our notifications up there, and there's some great little videos. Also look towards local media. Please, I would encourage all of you to lean on the local horticultural media in WA. There's a lot of people who are doing really cool things in their own time, a lot of podcasters, so get out there and watch that. We've got more information on the response at Kings Park on our website, and also for any botanic gardens on the line today, please keep an eye out for some of the training that we've got coming through began soon. Cool. 15 minutes.
Thank you very much, Chelsea, and thank you, Rohan, as well, for the passion and knowledge that you have both exhibited during those talks. The reason I've brought everybody on stage at once, we did think about doing this small group by small group, but I think there's a number of questions that came through. There's nothing, I think, that's going to be earth-shatteringly political or difficult to answer, but I think there's a few of the questions that were originally asked of one person, but they could well be a source of discussion and a little bit more ongoing than that. So I'm sure I've never had to tell any of the science team before, contribute, join in, keep that discussion going. When we need to quieten you down, we'll quieten you down. I'm going to start with Robyn because I know Robyn was dreading getting a question, so I thought I'd just start with hers to start with. Robyn, how should medication burning factor into PSHB management? And currently for all sites, including non-infected areas, we prune and leave branches on site for burning at a later date. Any commentary about burning?
Is this on? Yes. So essentially, to begin with, for our householders, don't burn. That's the key message. Leave your waste and on site for at least 6 months, and then when you are going to be transporting it, make sure that it is covered. When you're transporting it to a waste site, that is, make sure that your green waste is covered. So that is a very key message. We have been working with like camping and camping industry, caravan parks and the like, to promote people not to bring their own wood should they go camping. And the message is burn it, buy it where you burn it. Yeah. But in terms of mitigation burning, and I don't really have the answer for that. I don't know if you do, Vincent. Yeah, thank you.
Thanks. It working? Yeah. Thanks. I think the key message is that to remind people to comply with the condition of the quarantine area notice, and it's on our website where you can type your residential address to find out exactly where you fit within the quarantine area, whether you are in the management zone, in the control zone, outside of the quarantine area, and therefore which condition apply to you with regard to green waste, wood, any potential carrier of the PSHB. So I think that the message would be, if you've got an infestation of PSHB, yeah, it's probably not a good idea to burn the pile of wood, because that's going to encourage a PSHB to fly and disperse. Having said that, with the firewood issues, is that if you are buying seasoned firewood, so we're talking about something that's really dry, we know that the PSHB needs to have the fungus to survive and everything. So when we say do not move wood and green waste, the firewood that you buy at the shop in terms of the Jara, the red gum, the Mari, if it's well seasoned, the risk is managed by the fact that the PSHB does not live in dead wood for a long time.
Kirsty, through the WAARC partnerships, how are universities involving students and the next generation of our biosecurity experts in the PSHB projects?
So one of the criteria for the research projects was that they included PhDs and postdocs within the actual planning of each of those research projects. So of all of them, I'm aware of probably 2 or 3 PhDs that are currently engaged, and there will be more coming online as well. So certainly engaging them as much as we can in kind of discussions so that they actually understand the context in which they're operating, but also including their findings as soon as we can as well. So.
Cool. Tom, you've done a lot of local government and industry workshops at the moment so far. Can you summarise some of the feedback that you've been getting from those?
Yeah, a lot of it is probably, about, is this, I guess, some of the best practices that we give are obviously makes it sometimes maybe a bit vague. And so they probably want more input about how specifically they can deal with it, depending on who they are as a stakeholder. So, and how it actually affects them and what they actually need. So obviously we work close with, but internally, so obviously with Rohan, we've worked to Horticulture, they've done an amazing job of introducing us to other industries, and they, with the feedback that we've gotten even during the workshops and before, is that they want very specific, very easy to understand ideas about PSHB and how it works and how they can incorporate it into the way that their industry or their agency or whatever works. So if anything, the feedback has been generally positive, but I think that they definitely want to know how it affects them specifically and how we can fine tune it to make sure that it actually is effective management for themselves.
Cool. Rohan or Vincent, have you got anything to add to that from teams that you've spoken to?
I think that the one thing that stands out about this pest and the bug itself and the fungus is that it's just one of those things we heard today. It's really, really hard to kill. So these guys want definitive answers, and there isn't necessarily a definitive answer. And that's probably what's most challenging for all the growers. Some people are looking for a chemistry response. Some people looking for a biological, and there just isn't one thing that fits all. So this is I think really important message is guys, be open to the fact that this is going to be very different, and you're going to have to think about your situation and put it in. So the feedback I'm hearing is probably frustration without having a definitive answer, which is why we need to engage frequently and often so that we can provide the most up-to-date information we have, and then, as I said, learn the gaps that we haven't thought of ourselves as well.
Question that came through for Vincent, but I think this is a general operational question as well. How will the host list be maintained during and after the transitions management period, and how might stakeholders contribute and engage with reporting potential new hosts?
Okay, thanks. I'm having to have a start. So on our web page, so if you Google DPIRD and PSHB or Polyphagous Shot-Hole Borer, you will land on our web page where it's got all the information, including that list of hosts, the various susceptibility of each host, the amplifier list, and the like. As we heard this morning, as part of the response, we've added 130 new global hosts from memory. And that web page is, or that information is updated on a regular basis. Basically, every time the diagnosticians tell us that PSHB has been detected, in such and such new species, which wasn't known to science or to date, it's basically updated on our list on our website. So that's going to continue at the moment for the duration of the transition to management, because we've got the funding to do that. We've got the whole team. We've got 88 people from memory as part of that response. And therefore, that's going to be ongoing until at least December 2026. After that, that's the kind of work that is being developed as part of the future proofing part of the response, and basically the information that we are gathering in conjunction with talking to the international expert and everything, we will provide advice to government, and not just the WA government, but also the rest of the Commonwealth and the like. And therefore, that will inform what's going to happen after December 2026.
Thank you. We've had a couple of questions, one for Shannon and one for Francois, similar-ish. So Shannon, we had one that was from the Californian experience with respect to preferential sites of attack within tree architecture. Is there any evidence that more mature bark and wood chemistry offers differential resistance?
I think that, I wouldn't say that we haven't seen anything like more mature bark or anything like that. I mean, definitely younger trees that aren't as mature, really young saplings don't get hit. So they have to reach a certain age where they, there's good amount of functional xylem for the beetle to attack. So for us, I think it's mostly just having a good size and substrate. Similar to in South Africa, we see the beetle attack mostly on most hosts on the trunks of the trees, and they'll kind of move on to branches and things like that. And they will attack indiscriminately along those, but typically we see them prefer larger substrates first. So it's more about kind of host age and having a larger substrate to attack.
Oh, and Francois, same but different in terms of this question. What research-based evidence is there about how long a beetle can survive in harvested timber, either dressed or undressed? And is there any evidence of beetle infesting dressed timber? Treated, yep.
Yeah, so there's no research that I'm aware of that actually tracked how long they can still be active. So what I know is just what I've seen from dropped branches, and it's at least up till, well, depending on the environment. So it's in the tropical environment is about 2 months probably. Drop branches in which we were still able to find the eggs and the larvae in these pieces of wood. So what actually happens in the system, well, what we're picking up is that the beetle switches from the Fusarium fungus as main food source to the Graphium fungus in the drier wood, and that Graphium fungus can grow in dry wood. So the beetle, it's much like a farmer that takes a couple of things with it when it goes to a new environment. So it relies on these two fungi, not just the one. So about 2 months in the wet environments that I've seen. We haven't actually physically tested that. And the second part of the question, so a colleague of mine sort of reported the PSHB that actually drilled into his table while waiting for processing a sample in a laminator table, and the same thing started happening with me as well. So I think once they sort of enclosed in a captured environment and they try to escape, they'll chew their way through almost anything. But physically, they won't go into treated wood or dry wood. They won't colonize that at all.
And is there anything related to the volatiles from different tree species and the beetle response within those trees?
So I'm also, again, I don't think there's any research looking specifically at that. I think the very first indication that there might be something is what we're seeing with the forest trees being almost selected for some species. Most of the species on that list that I showed, most of them were randomly chosen. So there's a big part of random searching in the environment. They don't know any of these trees. They've never met any of these trees before in their native environment. So every single tree is new to them. But then it seems like certain species, they can select. How they do that, we don't know.
So Kirsty, this isn't actually a question, it's more of a comment. I hope you're keeping a list of things that haven't been researched yet. Where was I at? This sort of led on to a question that I had when Shannon was talking, but one of the questions here was, who's responsible for management in California and how's it funded?
It's pretty much up to the landowner that is responsible for land management. So whether it's a grower, an avocado grower, or somebody, or if it's depending on who owns the land. So U.S. Forest Service, or if it's a state agency, or if it's a private landowner or a nonprofit, it's up to them on how to manage. But we have, there is kind of a vested interest in wanting to manage it, appropriately. And there is a coordinated effort to communicate among stakeholders together on how to manage and what actions are taking place. So there's no directive. It's mostly kind of from the ground up that is coordinated with the state is basically helping to create the forum for communication among those stakeholders to do that.
So the question that I was scribbling down when you were talking was a little bit along those lines of Australia, as you'd understand, and you're both from very big countries as well. Big country, lots of stakeholders here. Are there any lessons that you would be able to provide us, or hints or ideas that you'd be able to provide us that helps with that communication, helps with that coordination across that whole range? Because if we had one fail in the system, then that's a weak point for the rest of the country in that space. So what's a lesson that you might give us in terms of that coordination?
Yeah, I think that having regular open communication, we have meetings on a quarterly basis with everybody, and those are open to the public. And so they're public meetings, and we basically talk through kind of the different priorities and action items, and then there's an opportunity for public comment as well. And so that kind of, I think that open engagement and input from stakeholders is something that is, I think that gives people buy-in to the process.
Francois, have you got anything to add in that space?
Yeah, I think from South Africa's point of view, things are a little bit different. In that, right from the beginning, we had to deal with more with all the different communities, and no real state action on that. There is some funding for some of the research projects, but no sort of effort. Nothing like we've seen here in Australia. I mean, what's happening here, it's spectacular. If we had that, we would not have had the beetle the entire country where it is now. So for us, we focus quite a lot on the arborists and the National Botanical Gardens as centers where we can train people that help to train others, so that there's regular workshops and things happening all over that we actually don't have to be involved in ourselves anymore. And there's also lots of these friends of park societies, and they often have meetings where you can actually speak to people in the public and train them. But other than that, there's one other place also, so the Grower Society, the Horticultural Society. Now that it's actually in the fruit trees, now people are interested. So there's these regular yearly forums for that. And they're also helping with some of the research funding now. So a lot of the chemical trials and things we do currently is actually through the funding from those industries.
We've got native trees and systems are heavily impacted in both South Africa and California, which is also the case potentially in WA. Do you have any advice for native system or tree health and mortality monitoring over time?
Oh, buy drones. I think that's the best idea would be to monitor it from the top, to see which plant species, which tree species are being impacted, and then ground truthing afterwards. And then build from there to see which species, how much danger there is, and then monitor all the environmental variables as well at those sites, compare it to sites where nothing is happening, to see if you can figure out why certain species are targeted in specific areas. And it's also important for the agricultural industry to understand which trees are actually contributing to that risk. In South Africa, in the stone fruit industry especially, it's mostly a problem, a sort of a management problem of what's around the orchards and not in the orchards. So reducing that impact.
I would just say, just to echo that, but also take advantage of the use of traps and have a trap network, in those adjacent to kind of your native vegetation, so that you can kind of track, where the beetle is relative to your native vegetation, and then follow that with visual inspections. And if there is a way to then also track abiotic factors and things like that, so that you can link that with what is controlling landscape spread as well, I think is really important. Having kind of at least intervals of areas where you are monitoring on a systematic basis, I think is going to be really key.
Chelsea, have you got anything to add to that from the Botanic Gardens space?
Yeah, just a little soapbox thing. The term tree is completely made up. So there's a lot of definitions for what is a tree. A lot of the time we think of it in quite that sort of colonial landscape context, where it's a single-stemmed plant with a nice bush of green on the top, and it's either above 3 meters or 5 meters, depending on how you want to look at your statistics that you're reporting on. What we're finding in a lot of our systems in WA is that things that are known as trees, the public might think of the tree outside their front house, out the front of their house. However, we're looking at long-lived woody plants that can be a lot smaller. So things that people think of as bushes or shrubs are impacted as well. And we're moving through those systems where it might not just be a single large tree. And so we need to be letting people know to be doing that monitoring and surveillance in some of those ecosystems that might not look like a typical forest or orchard. And there were areas as well that are quite concerning, especially through some of our riparian systems in the southwest. They might not be big trees. So just monitoring some of those species as well. Just getting I realized all day today we've been talking about trees, but we need to be talking about woody plants that are long-lived.
Thanks for that. Vincent, if I don't ask this one, people are just going to ask why I didn't ask this question. So I apologise. Can the team provide any insight into what post-transition to management will look like? What assistance will be available, for example, the department's diagnostic lab services, environmental biosecurity, regional surveillance, agricultural support?
Yeah, thanks. So I think I've already alluded to that. So as part of the transition to management phase, which is still nationally coordinated and nationally funded and nationally reviewed, we provide situation report and having a regular meeting with our federal and other state and territory and industry big body counterpart. We've got until December 2026 to basically collect all the information, gather a lot more information in terms of what's going to happen to some of the species. We all know that we stopped removing infestation within the management area of the post-metro area back in July 2025. So we're still 8 months into what's happened when you do not manage the infestation anymore within a specific environment, which is the metro area, right, of the management zone. So therefore, as we go, we're learning, we're collecting more information, we're having this kind of two-way street information session and workshops, and obviously we're learning a lot from all the various stakeholders as to what needs they have. For example, today that was a national workshop. We had similar workshop with state and territory counterpart and the like. So that's part of the transition to management. As I mentioned before, from a department perspective, we will be continuing to engage with the other state and territory and the Commonwealth and industry on the national level, at the state level. And we basically, as a department, will provide some advice, and it will be up to government to decide what's gonna, what kind of funding mechanism will be in place post the transition to management phase. So it's still early days, but basically at the moment, the quarantine area and the restriction will remain in place until at least December 2026. After that, it will be basically all the advice that we provide to government and up to government to, decide as to what's going to happen post T2M.
Rohan, I was just going to ask you to comment because Chelsea had her chance on the soapbox. It's all over to you.
There's no soapboxes here. For me, transition to management is taking it from a place where you've got a national response to you're managing it through the industry. And this one's a really challenging one because it's got such different values for different industries. But my advice from a horticultural point of view was going, well, what are the limiting factors for us to be able to doing that? What's the research that's already being done? We're looking at how it's spreading already. We're looking at what are those trapping techniques that can improve the trapping efficiency, effectiveness. Then it's probably the diagnostics. We've got this pest that we know that is not unique in its look. It's really hard to just look at it and diagnose it. What are the quick protocols that could be put in place where industry can get more involved in helping with surveillance post 2026? That's what I think we should be prioritising as a horticultural industry and investing in those solutions so that post transition to management, we are better prepared to manage it. And that's what T2M is designed to achieve. It's not something that the government will be going on continuing the funding, and that's how the deed works. It's there to respond to either eradicate or to take you through that transition to management. So I challenge industries to get involved in helping design what that needs to be and helping get us there at the same time.
We've been incredibly lucky over the last 3 days to have both Francois and Shannon here. They've been out and had a look at some of our locations that have been infested with PSHB. Have you got a closing comment from each of you for the people that are here today?
I think we've said what we need to say. But my best advice would be to keep on doing what you're doing for as long as you can, because it's obviously working. Then propagal pressure is being kept down. If you switch to something that you can't do that anymore, you're going to see a lot more infestations, a lot more serious infestations. South Africa is a prime example of that, and places in California as well. If you leave it, you're going to have problems. Yeah, that's it from my side.
Yeah, I would say that I think that the effort here, this is a really difficult pest to manage. As you have all, I believe, have come to really appreciate. This is not an easy pest to manage and to keep at bay. And so what the effort that has been put in and the resources that have been put into dealing with this beetle has been tremendous. And I think that Western Australia has done an amazing job of really keeping the population pressure down. And like Francois was saying, I think that if you just let it go, then that you're going to see a lot of damage and a lot of spread and a lot of trees dying. And so it is worth the investment of monitoring. If you were going to invest in anything, invest in monitoring your trees and invest in just those, those cultural practices. Don't dismiss those. Just to keep the population pressure down, that and monitoring and checking in with the trees. It's just an iterative process, but it is worth it. And if you keep doing that, then you can manage and tolerate the beetle. But it's important to do that and not just kind of throw your hands up and say, oh, it's here, it's going to spread, and there's nothing that can be done. So, yeah.
Thank you so much. I think we will give the speakers a chance to rest now. Thank you all very much for your contribution this afternoon and throughout the day. If you'd like to take your seats, and then we'll get Stephen up as well. Thank you. All right, I'd like to introduce Steve Nell, who is the Incident Controller for the Polyphagous Shot-Hole Borer response at DPIRD. Steve.
Thanks. Hi, everyone. You'll be pleased to know that we're on the home straight now. A few more minutes and we're done. First up, I would like to once again thank all of the speakers that we've had here today. We've had a huge amount of really interesting information, and certainly my team over the next period will be digesting a huge amount of that information. A lot of it, I'm pleased to say, validates what we have already done and are now doing. A lot of it is information that we will take into account to determine where we go from here. So I would also like to emphasise not only the speakers who've done a fantastic job, but to thank everybody else who has attended here in the audience in person, as well as online, if the online people are still there. We really appreciate the time you've taken out of your really busy days to come along and hear this stuff. And there will be more asked of you in the months to come. One of the things, so thank you all for that. One of the real issues that I'd like to emphasise again is the importance of providing feedback. A lot of the information provided by the response team, predominantly by Robbie and Tom, in relation to slowing the spread of the pest and in relation to capacity building, is fundamental to exactly the question people have asked, which is where we go from here. A lot of information will be coming out over the next months. A lot of that information, if not all of it, will really be asking all of the stakeholders, industry, institutions, community, and local government authorities to provide feedback. It's really no good, I'm stating the obvious here, but it's really no good at the end of this response for us to come to you as government and say, here you go, that's what you can do, we're out of here. What we really need is for industry to own this thing, and by industry I mean all of those different sectors. We need you guys to own this, we need you to have real, or to make real contributions to it. we will get information out there as a final draft, not as a final document. What we really are asking for is for you to come back and say, be honest for you personally and for your industry or whomever you represent, to say, yeah, this is really good stuff, or it's missing a lot, we need more of this, this and that. There are several conduits through which you can do that, partly through the workshops that Tom described in his talk. And if you need any more information about those, contact him. And partly through the information that will be distributed as drafts to selected members of the stakeholder community, if that's the right expression, to provide feedback to us to make sure that when we're finished, we've got the best possible product out there. What have I missed? If anybody needs any information at all, we have a web page. I always get that wrong. It's not a website, it's a web page. If you don't know where that is, ask us, we'll tell you. That will continue to grow as time goes on. It'll provide current information, and more importantly, it'll start providing more and more information about what we are doing. A lot of questions have come up here today and on the Tuesday meeting in relation to where do we go from here. There will be, among other resources, a calendar of events which will describe to everybody exactly where our various teams will be, what we're doing. And again, that isn't dictating just, well, it is saying what we'll be doing, but it's also open to suggestion. If anybody thinks, and we have received some good invitations from people saying, why don't you come here and provide presentations and so on. Trevor Ransom yesterday, on Tuesday, interstate representative of various horticultural industries suggested attendance and presentations at a workshop meeting, I'm not sure what it is, in Adelaide in a few months' time. We'll happily accept that, be there, make presentations to continue satisfying the tremendous amount of information that's being sought now from interstate as well as WA organisations. I'm not going to go on and on. I think that's pretty much it. The one final thing I'd like to say is, or to pass on, is to all of my team who've contributed a huge amount of time, energy over the last many months towards making these two workshops and the field trip yesterday, the success that I believe they had been based on what I've seen and the feedback that I've had. In particular, Louise, you're not getting away with nothing. I really want to thank our subject matter expert, Louise. She has been tremendously effective, enthusiastic and professional in putting this whole thing together. You've been tremendous, Louise. Thank you for that. It's been a huge effort. Thanks again, everyone, for the presentations and for coming along. And yeah, drive safe on the way home, Mr. Traffic.
And last time from me as well. As has been mentioned earlier, just as a reminder, reporting remains a mandatory legal obligation under the BAM Act. So please continue to use the MyPest Guide Reporter app for any suspect detections. It will help in that T2M phase and future modelling as well, and as making sure that we don't have detections go unnoticed. And apart from that, that is it. Thank you very much, everybody, for your attendance. Thank you for travelling here today, and safe trip home. Thank you.